Yeah.
Hello, good morning. Good morning, everyone. Okay, so I'm Jen Salem, the Plant Lab and Greenhouse Manager for the Regional Science Consortium. And I would like to welcome you this morning to the second day of our 21st Annual Research Symposium. So for those of you that don't know, the Regional Science Consortium is located in the research wing of this building, and we are comprised of 46 member organizations that support the mission of scientific research and education in our area. So please enjoy the following presentations, and if you have any questions or concerns, comments throughout the day, please see the registration table or any of our staff here in the building. We'd be happy to accommodate you. And I am going to introduce our session chair. Haley Dahl is the Regional Science Consortium's Education and Outreach Coordinator, and she will be moderating this session. You're welcome.
This clicker, this middle part is a laser pointer.
Okay.
And if you guys want to introduce yourselves.
Okay. Are we okay to start?
Yep.
All right. So my name is Kawan Santos da Silva, and these are my colleagues here. If you want to introduce yourselves.
Hello, I'm Madhav Alani.
I'm Patrick Singh.
I'm Dylan Crawford.
And I'm Peyton Gee.
We're from the University of Pittsburgh at Bradford, and our idea for this presentation was to create an experiment that can be easily reproduced by undergraduate chemistry students. And the idea of this experiment was to use a simple reaction method called Fischer esterification to create esters, which we'll explain later on, but they're simple aromatic compounds. And this compound right here is isoamyl acetate, which is it smells like bananas, and it's usually found in bananas and pears. And creating this compound can help organic students enjoy the process of these somewhat difficult chemistry mechanisms, and it makes it a little more fun and enjoyable for them to partake in. And then these photos are just some photos of us partaking in the lab as well.
Yeah, like Kawa mentioned, this is a common compound found in bananas and pears, and it's also used a lot in the industry, in the food, beverage, and fragrance industry, to kind of replicate the same smell. Fun fact, this is the same compound that's found when a bee stings you. You have this compound released, which acts as a pheromone naturally to attract more bees as well. The reason we're focusing on this compound and its synthesis today is because, like Kawa mentioned, we want to focus and bring some light into maybe possibly making this an undergraduate lab, and just to show students how simple like foundational chemistry topics can be learned in class and then replicated in the lab to create a fun, interesting, and easily recognizable lab.
Now, this photo right here may seem like a lot, but this mechanism is just explaining the mechanism of Fisher esterification. And so before we get into that, let me explain a little bit about what Fisher esterification is. It's a substitution reaction, specifically a nucleophilic azo substitution, which is similar to an SN2 reaction in which a nucleophilic or a compound that has excess electrons, more negative, attacks another compound, which you can see happening right here. And then the azo in this case just refers to the carboxylic acid that we used to start the reaction. And then Fisher esterification also, again, like I mentioned, needs a carboxylic acid, and then it's reacted with an alcohol, and that's what differs it from other substitution reactions. And then I put a star here in the mechanism, because there's a couple things that I want you to get from this figure here, is that without the sulfuric acid here acting as a catalyst, then this reaction cannot proceed. And that's a key thing about Fisher esterification, that needs a strong acid to act as a catalyst. And another thing that I want to point out in this mechanism is at the very end I circled the water here. As you can see, there's a double arrow indicating that the reaction can go forward or backwards. This is very important when doing an experiment like this, that to maximize the yield and the products, you have to understand that the presence of water and the ester product can cause the water to react with the ester, causing it to go backwards, which can really affect the yields in more industrial settings. So that's an important key point here.
Yeah, to briefly just talk about the procedure we use. We reacted our initial set of chemicals, and we refluxed it for about 60 minutes. Refluxing is just a simple process where the vapors that are released from the solution that's being heated are cooled down rapidly through cool water being run right by the vapors. This was then separated using the centrifugal and multiple wash stages and then collected. And finally, we analyzed our samples using GCMS and IR, which I'll pass on to Dylan to talk about. Thank you.
Basically the machines and the ways we have used to identify the compounds as well as the final product was GCMS as well as FTIR. These machines allow us to identify the compounds in multiple different ways. For GCMS, we use, first we insert into the machine, it will volatilize the chemical, become a gas, which is then pressurized using helium at 5 psi. This allows it to flow through the oven, basically volatilizing it, then entering the quadrupole, which will allow it to go to different frequencies and electricity, allowing it to separate the positive ions, allowing us to identify the compound and different chemicals that we use to allow us to see the final product as well as the previous things as well to make sure we have successful esterification. For IR, it allows it to use a IR light to pass through the chemical like a very thin film using salt plates because the banana oil was a liquid. Using that, it allowed us to basically vibrate the chemical at a very subatomic level to allow us to identify the certain frequencies to identify them as like common mixtures. And more on that, for the frequencies that we have got for IR was isomyl alcohol, ethanoic acid, and isomyl acetate. Here are some of the key peaks that we have gotten. And one of the biggest peaks that we have mostly identified was OH and the C to O double bond. For the next one, which will allow us to show, here is the IR sheet we have. So the key peaks as well as the C acetyl iron esterification would have been OH. Right here is isomyl alcohol and ethanoic acid. And up here it would be our isomyl acetate, which as you can see, we switched to a CH bond. This is one of the biggest things allowing us to see that esterification was successful in our experiment. And there have been less issues, errors overall. And for our GSMS, this is our gas chromatograph. This was a big one because we only had one big major peak. And going on from there, That led us to get our mass spectrum and we also use an SIT database to get the literature value and SID database. The nice thing about NSIT was secondary on the computer, so it allowed us to identify the compounds as well as anything that could have been there. But according to our chromatograph that we have gotten, right here, this is what we have, and then this would have been more or less the literature value. According to both them, this allowed us to basically allow us to see that it was a successful esterification and our mineral oil was successful. As well as we also had a more or less banana aroma. So it allowed us to more or less guarantee that our thing was successful. And for possible errors, I will pass that on to. Oh, all right, yeah.
So kind of going off what Dylan said, you know, it's very important to be using these types of instruments to further prove your successful synthesis of your product that you wanted. And talking about one of them specifically, we used FTIR. Obviously, FTIR is used to identify and study chemical substances by measuring how molecules absorb different types of infrared light. Every molecule has its own unique absorption, so you can definitely use that to tell what functional groups are present in a product. And further, you can also test the purity of your product that you did form. And this is very important because you can start ruling out different types of errors that could have happened during the experiment, and that's very important for when you're writing discussions and coming to conclusions.
So in any science type reaction, of course, there's going to be errors. There's some very common errors for this specific practice. One of them is time. You need to make sure you refit flux successfully for 60 minutes and make sure to heat just the right amount. If you don't heat it enough, as Kala said, the reaction is reversible, and the reaction could reverse back if it's not heated at a correct temperature, at a correct time, the amount. Another thing is not enough sulfuric acid catalyst can cause the reaction not to occur. If there is too much sulfuric acid catalyst, they'll actually create an odor. Normally it's a black odor, but if there's not enough of sulfuric acid catalyst, the reaction will not take place at all. And then also some things to note, product loss during separation can occur. If there's any impurities in your final product, your final ester product, that can cause some incorrect IR and GCMS data. Also, the excess water can also cause a reaction change and can also revert it back if there is too much water in the final ester solution.
All right, kind of just like to wrap everything up here. Yeah, we did come to the conclusion that we had successful synthesis occur, and we used obviously our different types of spectroscopy to confirm that. Using FTIR, we did reveal that there was a disappearance of the alcohol seen in the isoamyl acetate. It was not present, which further confirmed our conclusion here. And then using GCMS, we revealed that there was one single dominant peak, and that further confirmed the purity of our product. And obviously Dylan did perfectly explain what those graphs meant. And future implications, I mean, like Madhav said himself, really this lab was just trying to have undergrad organic chemistry students use different types of organic lab techniques like purification and interpretation of IR and GCMS analyses. And really it's just trying to bridge the gap between fear and inexperience. And basically this lab was allowing us to properly demonstrate the right skills and basically make a fun lab experience for undergrad students.
We have time to take a couple questions.
I've got one for you guys. Is your system sealed enough that moisture in the air wouldn't affect it? If you were in a very humid environment, would that affect things? Or is the system that you're doing it in sealed enough that it wouldn't?
No, I can partially answer that. It depends on how you do it. In the way that we did it, we're more just experimenting to create this lab and see how it works and the efficiency of it. But if you are trying to make it more efficient, there are certain things that you can do to improve it. There's this tool called a Deep Stark apparatus that can really help with removing as much water as possible. And that has shown to really improve the yield and purity. So yeah, like I'm thinking more industrial settings and more advanced labs that would use techniques to improve water removal.
Any other questions?
So in terms of teaching this to undergraduate students, what do you think would be the biggest challenges for them in the whole process? Like what would be the hardest thing for them to either understand or to do in the lab?
I think the hardest may be like GSMS, because I know for us that was one of the biggest hardest things to understand, like how it basically converted it to the graph and understanding the graph, because that took me a couple hours at least to understand like how it's supposed to get even working equipment, because we used pretty old equipment like back in like the 1900s. That it mostly if you're using more modern ones like auto sampling. Honestly, if you're gonna do this with undergrad students, I would say if you're more college has it, use this auto sampling, have the teacher show the kids how to use it, because as well as the method can also be very effective. Because for us, we had to make sure it was at 40 initial, then 250 10 grades per minute or per second. This allowed it to easily identify the sample. Also, you also need delay as well. So it depends on your delay, depends if there's any errors or any impurities. If there's impurities, it will catch it. If you need to, you may have to redo it. But if you're going to be able to do an undergrad student, there's probably more than one, so you'll at least get one or two samples that will be guaranteed good. So.
Also, like, make sure you help these kids interpret what they're looking at too. I mean, you're gonna see a graph on the screen. A lot of undergrad kids are gonna be intimidated by it because they don't know what it is. But obviously, us, like our teacher, he showed us what it really meant, which was really important.
This level will mainly be working with the teacher as well as students, and finding time with individual to explain if they have any questions.
On the contrary, the actual lab process is rather simple. It's only setting up the reflux, so when it comes to the actual lab, students shouldn't have too much trouble. And like everybody's mentioned right now, it's really just the identification and the analysis part that will take some more time, just further understanding of the topic to really just get a hold of.
As well as maybe assuring them that even if it's a little bit of sample, it's still fine. Because I know with us, I was even worried that there's such a little bit of sample. It's like, did we actually do it right? Was it okay? But I know once we did the GSMS specification, we were like, it's good, we're good to go.
Good experience for them.
Any other questions? All right. Happy to introduce Isabella Udoko.
Good morning, everyone. My name is Dr. Udoko. I'm a professor at Ghana University. I'm not just at Ghana University, but I teach in the psychology program, and I'm also a professor of organizational learning and leadership. I want to introduce my presenter today. She's actually the one presenting. I'll be here to answer questions and to offer a little bit presentation support. No, we're not sisters. She's my daughter, and she's a freshman at Penn State University. She started exploring this research. In fact, it's still in the early phase of development. As she came into teenage years, started the adolescent period, going through puberty, she noticed how susceptible her cycle was to large changes such as intense sports and activities. She was an athlete when she was in high school, and she asked a lot of questions like, why does she feel this way, or why does she have breakouts during certain parts of the time of the month? And raising her alongside her brother, who's also a teenager. She had interesting observation, like why her body would respond differently versus his body in regarding to certain situations or like stressful stressors or stressful situations. So she started doing those early research from about age 12, and 4 years later she has developed it into what we will hear about today. So I want to take the opportunity to turn it over to her now. Isabella?
Well, thank you everyone for taking the time to attend my presentation. Today I'm going to be talking to you about the secondary immunology of stress, or PNI, and stress regulation or cortisol regulation in early adolescent girls. So I'm going to start off with talking about what PNI is. I'll give you a background of my research, the rationale and significance behind it, the theoretical background, a literature review summary, my research question and hypotheses, my methodology, research implications, and I'll give you time for questions as well. So first, what is psychoneuroimmunology? Well, it's the study of the brain, the immune system, and behavior, and its interactions with each other. It started off with a psychosocial model and then changed into a biopsychosocial model. The model is a framework that is used to better understand the effects of biological, psychological, and social factors on health. This model here is a representation of a visual representation of the biopsychosocial model, showing the relationship between biological, social, and psychological stressors. Research has shown that adolescents, I'm not sure if I can, let's see. So that is the biopsychosocial model. It shows you the relationships between biological stressors, psychological stressors, and social stressors. So that would be disease and bodily health for biological, aspects of mental, emotional wellness for psychological, and interpersonal and social interactions with your community and other beings for the social. So, adolescence, research has shown that adolescence has been a very critical period in time for development. The study of PNI has originally stemmed from the idea that psychological stress can very much influence your nervous system. It started off with a primary focus on the brain and immune system signaling and its effects on various psychological factors on immune system health. More recently, PNI has changed into studying how the immune system affects brain functioning as well as psychological processes. To better understand how PNI works, scientists started off with studying rodent maternal separation. The researchers found that pups that grew up with the nurturing of their mother were less susceptible to stress compared to pups that did not grow up with their mothers. Later on, stress evolved into sex differences in rats and how they interpret stress. Researchers found that female rats were more susceptible to stress due to the presence of estradiol, which is a female estrogen hormone, compared to male rats who had testosterone, which ended up suppressing that expression of stress. On the other hand, we've been able to find that these key differences are very important to understanding PNI, and which is why we're going to focus on girls. So, whenever an individual is stressed, the sympathetic nervous system is initiated, and which leads off into the hypothalamic pituitary adrenal axis, which also gets activated right after that. So the HPA axis, or what I just explained there, is basically what happens when you are hit with stress. It produces cortisol, which allows your body to realize that, oh, I'm in the process of being stressed out, and it causes a constant reaction until you're able to relax and your parasympathetic nervous system takes over. The SNS, which is very important to this, without the SNS, the HPA axis will not be initiated. It starts off your fight or flight, which then leads off into your HPA axis being initiated, ending up with the ultimate release of cortisol. So it has been found, It has been found that stress and social media exposure in teens have greatly increased post-COVID due to adolescence being a critical period of adolescent development. Research has also found that early adolescents have been more susceptible to HPA reactivity, also due to the fact that adolescence is a critical period of development. But why focus on girls? Well, research shows they have higher levels of internalization, knowing that they are more sensitive to emotions compared to males. This is, again, due to the fact that we have estrogen, which just heightens those effects of outside factors on females, compared to men or males who have the presence of testosterone, which dampens those reactions. But why focus on specifically early teen girls is because of the fact that early teenagers, you're being flooded with completely new hormones such as estrogen and testosterone, which makes you a lot more susceptible to falling prey to stressors, which can lead to later on more health issues psychologically and immunological. Compared to late adolescents, they already had a long period of time where they've been dealing with these hormones, and they are also engaging in maybe sexual activity, they have work, they're in school, maybe in college or even late high school, and they are used to the effects of their reproductive hormones. So, when the body perceives a stressor, the hypothalamus signals the HPA axis, which is this whole thing here, this whole cycle. This process begins with the secretion of corticotropin-releasing hormone, or CRH, then stimulates the anterior pituitary gland, known as the master gland, to release adrenocorticotropic hormone, or ACTH. ACTH then circulates through the bloodstream to the adrenal cortex, which ultimately ends up in the release of cortisol. So when cortisol is released, it slows down this process again, which is shown here by the negative feedback loop, telling the hypothalamus that it can slow down in this production of CRH and telling the anterior pituitary to slow down in its production of ACTH. When HPA activity gets too high, cortisol levels rise to a point where this negative feedback loop is tapered off and the reactivity from the HPA axis dulls. So, cortisol is a biomarker of chronic stress. Hair cortisol reflects the cumulative amounts of cortisol over long periods of time, hence why it is used for studies that are looking at the amount of cortisol, a good amount of cortisol over a long period of time. There are studies that have shown that there is a clear relationship between elevated cortisol and academic pressures, family conflicts, and peer victimization. There is also a clear gap in the amount of research focusing on early adolescents, and even more a gap in focusing on early adolescent girls. So here's my research question and my hypotheses. My research question is, does self-reported stress predict hair cortisol in girls aged 11 to 16? My first hypothesis is that higher levels of perceived stress will be able to give off higher levels of hair cortisol. My second hypothesis is that these associations will remain after adjusting for factors such as age, body mass index, pubertal stage, recent illness, and hair treatments such as dyes, keratin, and other treatments that you may think of. So my participants for this study will include about 60 girls from ages 11 to 16. My team will be recruiting girls randomly from local middle schools, and my measures will include the Perceived Stress Scale, or PSS-10. I'm just using that scale because of the fact that research shows it has good validity and it is a good measure of stressors in girls, and it usually is around 10 questions. I'm using hair cortisol concentration, in which 3 centimeters of a hair sample is equal to about 3 months of cortisol. And then I'll be controlling for factors such as age, pubertal stage, and socioeconomic status. So my procedure will come about with starting with recruitment, as I explained on the previous slide. My team will be getting informed consent from parents and child assent as well. The hair samples will be collected from the posterior vertex, and there will be a questionnaire. The posterior vertex is basically the spot in the back middle of your head. It is not as affected by the outside things, so air, brushing, touching your hair, it's not as disturbed compared to the rest of your head. or head. All the data will be de-identified, which means I will not be able to get IP addresses, location, anything that has to do with the person giving answers to anything at all. And then I will be receiving ethical approval from the Institutional Review Board. So this is just a quick, brief overview of the methodology, starting with the, wrong thing. Getting the hair from the posterior vertex, and then my team will be measuring. So we're gonna need the hair cortisol closest to the scalp just because it's accumulation of hair, and we all know hair doesn't grow from the bottom up, it grows from the scalp. Then we're gonna weigh the samples just as another control variable. We're gonna extract the cortisol using methanol, and then we're gonna be able to analyze our data using the liquid chromatography mass spectrometry or an enzyme-linked immunosorbate assay. So the implications that my research may have on PNI and theories is it will advance the field of psychoneuroimmunology as a whole. We will be able to get more information on the linkage between psychological stress and biomarkers, and even more in the aspect of girls. We will also be able to support the fact that adolescence is indeed an incredibly vulnerable period for stress regulation, and we'd be able to find out ways that we could prevent the effects of stress or help in reducing stress. The practical implications for my research is that it simply shows the risk pattern for girls, making it a little bit more predictable and a little bit easier for us to help and to intervene when needed. It would also be able to support early intervention, encourage school programs that may help, again with intervention, and educate families on this problem so that they know what's going on in their child's life. So, any questions?
This question may be a little outside of your scope, but I don't know how common it is, but what if there's adolescents going through hormone treatment, be like male to female or such? Let's go like an example of male to female and they're getting those hormone treatments. Could their levels of stress be comparable to a cisgender female woman or female adolescent?
Okay. So it would be a little bit challenging because of the fact that there's still levels of testosterone within the body. So until those levels of testosterone taper off to a point where it's a baseline of a cisgendered person, then it would be a little bit more challenging to say that the stress is related to the amount of estrogen they're being pumped with. Yeah. Any other questions?
So when you collect the hair, I thought it was interesting that you would weigh it afterwards. So are you going for like a total amount? I'm just thinking different hair, like some people's hair is really thin, some is really coarse, like mine, so I'm assuming like the more coarse the hair is, of course it would weigh more, but it would have more potential to accumulate cortisone markers.
So it would be a little bit challenging to think about that, but I did learn that it also has to do with different hair colors and different hair types. Yes. So that would also have to be another control factor, the different types of hair, because thicker hair is more likely to contain more cortisol compared to thinner, more straight hair. So weighing it would have to be within its own control. So curly hair being weighed to a certain point, straight hair being weighed to a certain point, wavy hair. So it would be within a different frame.
Are you going to note that in your data?
Yes, I will.
Okay.
Yes,
Yeah,
I will.
interesting. Thank you.
Of course.
Yeah, great question. And we want to have like a minimum so we could get those estradiol or those markers extracted from them. So certainly the variations in hair texture is going to affect that, so you'll control for those things too. Thank you. Question?
Oh, we'll start with you. If you're back.
During your extraction, you wanted to use methanol. Do you know if you have tested other solvent and what temperature? The solvent you use, the temperature you use have effect on the amount of the biomarker you extract.
Okay, so I've looked at a few different research papers, and one of the most recent ones that I looked at had looked at accumulation of different experiments using hair cortisol specifically, and it seemed that using methanol was the best one to make sure that the amount of hair cortisol that would be found isn't completely thrown off by using methanol. So it'll be best to use methanol in this case, just to make sure that those measurements are more accurate compared to using different other other chemicals.
Are there other chemicals that's used?
There are other chemicals that are used. I've seen a few that they use pure isopropyl alcohol. So, but those ones are usually dries it off to the point where it's like there's no cortisol at all. So, or very low levels of cortisol, which wouldn't exactly be accurate.
Okay, so the methanol is the ideal marker.
Yes.
To keep those cortisol indicators for testing.
Oh, and you had a question as well.
Yeah, I was going to ask, are you going to take any samples from adolescent males as a comparative study?
Not for this one. Not as yet. No.
And certainly it will be interesting to make those comparisons to reiterate the fact that women or female systems look different and their reactivity certainly looks different. So I think that would be good for future studies. Thank you.
Any other questions?
Okay.
Thank you.
All right, and for our last presentation for this session, I'll now introduce Nardos Bisrat.
You can advance the slides using this and this.
Hi, good morning. My name is Nardos Bisrat. I'm a biomedical engineering student at Gannon University, and today I'll be presenting on my project at Lab on a filter immunoassay microplastic detection. So to begin with, what are even microplastics? So microplastics, they are plastic particles that are less than 5 millimeters. They come from a breakdown of larger plastics or microbeads in products. They can be found in water, in food, in air, even in our blood. So there are common plastics like PE, PP, PVC, and so on, but what we're focusing on in my project is polystyrene microplastic, which is widely used in consumer products, packaging, medical devices, and constructions. PS is short for polystyrene. Is highly stable and it's resistant to degradation. So that leads to health concerns. So why are we even concerned about microplastics? So microplastics, they can enter in our body through injection, inhalation, or skin contact. That may cause inflammation and oxidative states. It can disrupt our immune system, and it could be a potential toxic chemical leaching. And that could possibly bioaccumulate in our tissues, so it could accumulate in our organs and even in our bloodstreams. And that could cause the long-term exposure risk is still being studied. Current detection techniques and limitations. So we have current detections techniques like the Raman spectrometry, the FTIR spectrometry, and the pyrolysis GCMS. However, all these techniques, they do detect microplastics, but they do have limitations. Some of the limitation is that they're a very slow process, so it can take a very long period of time to detect the microplastics. They can be really expensive. The equipment used for these methods are really expensive. and they might need trained, experienced person to run this process. So it's not really ideal for clinical diagnostics. So the goal of our research is to develop a rapid, low-cost, and easy-to-use method for detecting microplastics in liquid samples. And the approach we used is a lab-on-filter colorimetric immunoassay. So we used primary antibodies, which recognizes our microplastics. We used secondary antibodies that are linked to HRP enzymes, which generates the color, and that gives us signals intensity, which corresponds to the microplastics amount that we have. The advantage for the approach we used is that we don't need any expensive equipment. It's a fast process and it's scalable to multiple samples, and there's a future potential in clinical diagnostics, so it can even be used to study human bloods. So the approach we used is ELISA immunoassay, which ELISA is short for enzyme-linked immunoabsorbent assay. And what ELISA is in general is it tests the uses of antibodies to detect a specific target. So binding is detected through enzyme reaction, which creates a color change. That is what we study for the intensity of our microplastics. So if we have more color change, that means more target is present. So our system, it adapts the concept of ELISA to detect the polystyrene microplastic by using antibodies. So this shows the workflow of our experiment, which is our procedure. What you see over there in yellow, that's the filter we used. So what we first do is we inject a gelatin buffer onto the filter. What that does is it creates a layer on top of our filter so we can collect the microplastic beads. And at the bottom, we first made a solution with our first antibody and our microplastic, the PS microplastic. So similar ways how antibodies work in the human body, the first antibody, it recognizes the antigen, which is the target. So in the body could be like bacteria and viruses. In our case, the antigen is the microplastics. So we push that solution onto the filter, and as you can see here, the first antibody and the microplastics will be on the surface of our filter. We continue with the washing process, and the reason we use a washing buffer to wash it is to remove the first antibody and the plastic beads that are not attached onto our surface. We continue with this second process where we put second antibody on top of our filter. And the reason for that, again, similarly how it works in our body, the second antibody, it recognizes the first antibody, so it attaches to it and kind of creates a chain where we have the first antibody, the second antibody, and the microplastics. We continue washing process for similar reasons. Then at the end, we will use a chemical called TMB. What TMB does is it reacts with enzymes and it changes the color. Since our second antibody has enzymes in it, it will change the color, and we can use a device that can give us the intensity of the color it has. Using that method, we are able to detect microplastics. So the optical density measurement is what we use to detect these microplastics. So we use a molecular device called the SpectraMax microplate reader that uses to measure the optical density at 450 nanometer, and after the TMB color change reaction happens. So the amount of the microplastics were captured on the filter. So if we get higher OD value, OD is short for optical density, the that means we get a stronger color signal. And having a stronger color signal, it means more microplastic has bounded to the antibodies. We also did an SEM characterization. SEM is a device that is called a scanning electron microscope that helps us study the morphology of our filter. The image you see over here is after we put the microplastics. So the circle white beads are microplastics that were captured on the filter. This image over here is just the filter, so this image clearly shows us that the filter has captured the microplastics. And the membrane we used for the filter is PES, which has a cut size of 0.2 micrometer, and the average retention ratio for our 2 micrometer microplastic beads is about 99%, and that is shown on this image here. So when we did this experiment, we first did optimizations for the primary and secondary antibody. That means we had to decide how much volume we had to use and how we could go on about this for our project. So our first optimization, as shown in this graph, is our first antibody optimization. We had to decide how much volume we needed to use in order to go on with our experiment. We did different experiments using different volumes, and as shown in the graph, it clearly shows that 2 microliter of first antibody is ideal for our project as it gave us the highest OD value. And on the graph here, we did a comparison for how much dilution factor we need for our second antibody. We tested different dilution factors as shown here. We did 15,000 times, 20,000 times, and we compared with the microplastics and without microplastics. As you can see here, most of it has a higher value when the microplastics were added, which is a positive result for us. But however, we were still comparing other values, so we decided to go on with 20,000 times dilution for our second antibody since it had a better signal with less background noise. We also did washing optimization. As I've shown you on the procedure, we used a washing buffer to take away the unbounded antibodies. And the washing surfactant we used is called Twin 20, and we had to decide what concentration to use for our project. We tested different concentration levels as shown on the graph, and we decided to go on with 0.1%. Even though 0.1% is a little less than 0% and 0.05%, it still gave us a better result since it had less error than the other percentage. And then we also did a washing buffer, used a washing buffer after adding the second antibody. And we tested how much volume we needed to use after adding the second antibody. And with the results we got, you can clearly see that using 6 milliliter of the washing buffer, it gave us higher results on all concentration beads that we used. So we decided to go with 6 milliliter of our second antibody. On this figure right here, it tells us the standard curve showing logarithmic relationship between our PS concentration and OD value. As you can see here, it has a linear increasing as the concentration of the microplastic increases with our OD value, which clearly states that when we have higher concentration of beads, the OD value will also increase. However, if we have, like here, how it stopped increasing, if we have a very large amount of microplastics, it could also be a bad thing since it will give us errors due to high signals in our experiment. but the limit of detection is 1.6 nanogram per milliliter. And what that means is the smallest amount of microplastic beads we can test is this number, which is really ideal for human, in the human blood, since we could have a range from 1 to 7 nanogram per milliliter in the human blood. So this test is ideal to proceed with testing it using actual human blood samples. So in conclusion, we developed a lab on a filter immunoassay for microplastic detection, and the sensitivity, like I've mentioned, is down to 1.6 nanogram per milliliter in microplastics. We used a low-cost reagents and like really low-cost equipment, and we were able to quantify colorimetric signal based on the antibody bindings, and this demonstrates a future potential to be used in clinical testings, like I've mentioned, human blood samples. So for future work, we will work on detecting in a spiked PSMS from human blood samples, and we will use red blood lysis and purifications to do this method, and the approach we used will provide versatile tools for detecting microplastics from clinical and environmental setups. All right, thank you. If I have any questions. Yes.
So the microplastic beads you used, you said they were 2 micron diameter?
Yes.
Where do you get such small particles?
You can purchase it like from different companies that sell like chemical reagents, or industries provide those microplastics. Same with the first antibodies and the second antibodies we used are also like raised in the labs. They're not directly from human blood. They're raised in labs with, the first antibody was raised in a rabbit, which was able to detect microplastics. So we purchased all those equipment so we could test our experiment. Yes.
So for those beads under the SEM,
Mhm.
what type of plastic did you say that was again?
For the filter,
Yeah, for the little beads under the SEM filter.
we used a PES for the filter, so the material of that filter is called PES.
So for different types of plastics, will it, like at that size level, for example, like under the SEM, would shapes be different, or would they mostly be bead-like?
For our experiment, we only tested polystyrene microplastics, so I can confidently talk about that one since I'm not sure about other microplastics, but I believe it would be different since the morphology of different plastics could be different. Even for the filters, when we first did the experiment, we tested different kinds of materials for the filter. Like we tested nylon, we tested other materials too, and the morphology for these filters were different. And the best approach for us was using PES. That's why we used it. Any other questions?
All right, so that will conclude our first session of the day. We are going to take a kind of extended break because for session 2, our first presenter will not be attending today. That being said, we do have some high schoolers in our crowd today, so if any of my presenters are sticking around and kind of want to chat to them, if you guys have any questions, it would be a perfect opportunity to kind of chat with them. But yeah, feel free to walk around, get some refreshments, and we will reconvene at 11 o'clock.
Yo. Yeah.
Okay. Hello, everyone. Good morning and welcome back. Hello, everyone. Can we have our seat, please? It's 10:59. The next session will start at 11 o'clock. Briefly before we start, as we can see, the first presenter is not here, so that's why we have delayed it to this time, starting at 11. We have 3 people in this session that will be presenting. We have 1 student, and the remaining 2 people are not students. So I want all of us to pay attention to them. And our first presenter today is Cassidy from the University of Pittsburgh at Bradford. Cassidy will be presenting on investigation of mist pollutant degradation. Cassidy, you can come for your presentation.
Our presentation is based on investigating the potential of using photocatalysts for pollutant degradation, specifically crystal violet dye and amoxicillin in UV light.
So aquatic pollutants. So water pollution poses a significant threat to both aquatic and human health. Aquatic pollutants often originate from industrial, pharmaceutical, and domestic discharges. Some examples include leachate, which is basically a toxic sludge formed when liquid moves through solid waste, and the improper disposal of textile waste and medicines, as well as medicines entering water systems through human excrement. So the improper disposal of household chemicals and quote-unquote flushables would also qualify for this. Aquatic pollution threatens the health of aquatic life, potentially causing gill damage, fin and tail rot, and the depletion of dissolved oxygen. Medicinal pollutants can induce microbial resistance through creating selective pressure, which is basically just natural selection, and the facilitation of genetic exchange. This problem is best treated by prevention and treatment in water plants, which calls for the development of catalysts that are capable of degrading pollutants that can easily be integrated into existing plant structures. In this project, we focused on catalysts that could be integrated into existing UV light treatments using photocatalysts to degrade these pollutants. So to start, we have amoxicillin. Among possible pollutants to be degraded is amoxicillin, which is a semi-synthetic penicillin-like antibiotic that is used to treat a broad range of infections from respiratory to ear infections and others. So this pollutant most often enters the environment through human excrement after being used as a treatment. Amoxicillin inhibits photosynthesis processes in cyanobacteria, basically by hindering the electron transport in photosystem 2. And this also, furthermore, it harms bivalves, which are just clams and oysters and mussels, by altering their immune system and microbiome, which can extend damage to human health if consumed, if we consume these things raw.
Yeah, they also have high potential for antimicrobial resistance as from their purpose as an antibiotic. Basically, this triggers a stress response that extends and creates a natural selection system that increases the mutation and resistance to antibiotics, which can harm human health.
So crystal violet is used as a biological stain in dye and inks, paper, and textiles. It can also be used as an antiseptic. It usually enters water systems from industrial runoff. It can cause tumors, disrupt the endocrine system, and cause cell membrane damage in fish and invertebrate. And its dark color blocks sunlight and makes photosynthesis in algae pretty difficult by blocking that light. Crystal violet toxins can also reach humans through the persistence of metabolites in the fatty tissues of fish when consumed. So for current research in amoxicillin and crystal violet degradation is focused on the usage of photocatalysts. as we have proven, or as it has been proven that it is very difficult and very efficient. My fault. Very efficient and relatively simple method of treatment. Studies focus on degradation in visible light, using a variety of catalysts and enhancements, and degradation in different pHs. So it was found that degradation is overall faster and more effective in UV light. Titanium dioxide, biochar, and other oxides are effective in different conditions and wavelengths. Hydrogen peroxide and inorganic salts enhance degradation, and for a more stable and safe degradation, a neutral pH would be best. So expansion on current research. I would say, however, current research most often shows pollutants are in pure environments, which is not realistic to environmental settings. Pollutants can inhibit degradation of itself and other pollutants by blocking light and creating matrices in solution. So the primary photocatalyst chosen for the experiment was titanium dioxide. It precipitates easily and is non-threatening to the environment, and it is pretty stable. So we chose to dope our titanium dioxide with combinations of copper, which increases light absorption, and activated carbon, whose pore size and ability to efficiently absorb pollutants make it an effective site for photocatalysts to occur. So we chose to create 2% and 5% copper doped titania, 75% and 80% activated carbon doped titania, 2% copper and 80% active carbon doped titania, and then 5% and a 75% active carbon doped titania. So this catalyst works by pulling pollutants onto its surface and transferring an electron to its conduction band when exposed to the light. This will create a radical cation. This cation goes on to create a highly reactive radicals within water and oxygen, which degrade the pollutants overall. So the products, crystal violet degrades into carbon dioxide, water, and nitrate ions. And amoxicillin degrades into mostly carbon dioxide and water with trace amounts of sulfate, ammonium, and nitrate ions. So as for hydrogen peroxide, we also chose to examine how hydrogen peroxide enhanced this photocatalyst. When combined with titania, it acts as an electron scavenger and ensures that the electron does not recombine with its electron hole, overall slowing down degradation. And it could also help create the reactive species that aid in degradation.
Alright. So to prepare this catalyst, we first took pure titanium dioxide in powder form, copper nitrate, and then activated carbon, which we synthesized from plantain peels, treated and heated. From there, we impregnated the molecules in 2% ethanol water solution to be able to effectively impregnate the titanium dioxide with the activated carbon and copper nitrate. It was then dried in a low temperature oven overnight to remove the solution, and then calcinated at 350 degrees at 350 degrees C for 2 hours to change forms and better pore size. From there, the photocatalysts were characterized using SEM imaging to determine the size of the pores in the different catalysts and the consistency of impregnation across the catalyst. And then XRF and elemental analysis was used to determine the effectiveness of the doping and make sure the percentages listed were accurate to the method used. From there, to show the degradation mechanics, 0.1 gram of photocatalyst and 0.1 milliliters and peroxide were added to 100 milliliters of pollutant at 10 ppm. We chose 10 ppm because it was most effective for analysis. Did not require dilution or anything. It was mixed and stirred using a mercury lamp to irradiate it, and then with samples being taken at regular periods to be able to examine the rate log. the absorbance of the samples were determined using UV-vis spectrophotometry to determine the percentage degraded at each time interval. Then also trials were performed without a catalyst present and without hydrogen peroxide for comparison as to their efficiencies. First, showing first the effectiveness of visible light without the catalyst. With hydrogen peroxide, only amoxicillin showed significant degradation and only to about 60%. And then the other one showed no significant degradation or not, and any degradation was inconsistent. And without hydrogen peroxide, there was very little degradation, and in fact increased over time just from matrix effects. In UV light, however, there is some effectiveness in degradation, specifically in mixture crystal violet, with a little bit of degradation to about 75%, and amoxicillin had very bad matrix effects with just hydrogen peroxide and UV light, as it's not more as efficiently degraded as crystal violet interactions. Without hydrogen peroxide, crystal violet showed pretty significant degradation to about 30% and 42%, showing it's effective with just UV light, but it's enhanced by these catalysts. Out of these things, the most significant rate law was the crystal violet, and without hydrogen peroxide, with a rate law of about 1.1879% degraded each minute. With the addition of the catalyst, 2% copper titania showed inconsistent results, and 5% showed minor results but consistent. 75% copper activated carbon titania showed very significant degradation of mixture crystal violet and overall good degradation, and 80% showed less degradation but more consistent. The 2% and 80% showed inconsistent degradation but very effective in amoxicillin, and then and same true for this 5% and 75%, with pure crystal violet also having a significant degradation. With the rate laws, the most significant ones for amoxicillin was the 2% copper and 80% activated carbon. For crystal violet, it was a 75% activated carbon. For the mixture, it was the 80% activated carbon. And for the mixture CV, it was the 75% active carbon. From this, it can be determined for pure, without hydrogen peroxide, catalyst of pure activated carbon are optimal. As this is given here. The most overall factor overall without the hydrogen peroxide is the 75% activated carbon, performing very well in the crystal violet in both scenarios. With hydrogen peroxide, the 2% copper titania showed much more consistent degradation and overall lower. With 5% it was less consistent, but still overall lower. With the 75%, crystal violet in a mixture had very, very significant degradation in both 75% and 80%, and overall improved degradation for crystal violet. And then the mixed copper and activated carbons, crystal violet continued to perform very well and joined by amoxicillin, and then overall degradation in the 5% and 75% improved with the addition of hydrogen peroxide with the addition of hydrogen peroxide. With the rate laws for this, as you see, overall the degradation rate increased with the 80% performing overall well. The most effective for the amoxicillin was the 80%, which is also the best for the crystal violet in a mixture, and that crystal violet in pure form is best improved by 2% copper doped titania, and 80% activated carbon and 2% copper perform very well with amoxicillin in a mixture. With the addition of hydrogen peroxide, the 80% activated carbon doped titania performed the best, and the most effective without and with hydrogen peroxide was the 80% titania, followed by the 2% copper, 80% activated carbon to titanium. From that we can conclude that overall 80% the addition of activated carbon to titania is effective at degrading the pollutants present, especially in a mixture with copper performing well in specifically amoxicillin environments, partially because of its effectiveness in light absorption. This can be applied to ongoing research as it's very effective at absorbing visible light. So, if the implementation of more catalysts more pollutants like radiant visible light can be incorporated. We still need to complete SEM images of catalysts to examine the pore size, and then because expecting an hour to degrade pollutants in water treatment plants is unrealistic, determining rate laws in 5 minutes and 15 minutes would be overall effective for determining the implementation of these into water treatment plants. Any questions?
Any question for the
Did you try any other variations on biochar? I know biochar is used for all sorts of environmental uses.
No.
I'm just curious.
We just use an application of titania. We tried to do different implementations of how much, but overall, if we did a bigger range, it would be more effective. But overall, it just absorbed the catalyst, and I think created a better way for it to interact with the titanium dioxide by being able to absorb into a closer range instead of in solution.
Yeah, I think the students before them tried to use different vouchers. One of the reasons why he decided to use plantain fuel was just because of the availability.
Yep.
Do you know if any of these compounds are currently being commonly used to treat pollutants, or is this all novel?
Titanium dioxide was pretty common, but I don't think they're widely implemented into water treatment plants yet, just because of research and also just environmental analysis of each pollutant in each environment is not as widely applied yet.
Thank you.
Thank you. Okay, we go to our next presenter, and that's Dr. Fred. Dr. Fred is from Presque Hydro, and he'll be talking on the monitoring and management of cyanobacteria, HAB, in shallow and near shore area of Presque Hydro. You have the floor, Dr. Fred. I have some stuff here.
All right, good morning, everyone. All right, so if you're not familiar with cyanobacteria, they're essentially a type of algae, and even though I call them algae, technically they're bacteria that act like plants. They're the ones that make the nasty, it looks like someone spilled green paint on the surface of the water. They can make the water look bad, they can have bad taste and odor problems, but one of the main concerns with cyanobacteria is they have the potential to produce cyanotoxins, which can make people sick, can affect wildlife, livestock, and pets. Dogs are very susceptible to cyanotoxins. We do a lot of monitoring and management for the lakes in Central Park in New York City, and invariably in September, October, you see in the New York Times a dog dying because, you know, dogs outside running around, they get in the water, they get out of the water, they start licking themselves, they're very susceptible to many of these cyanotoxins. So a lot of the stuff that I do is a lot of lake and watershed management, and so a lot of it is to reduce nutrients going into a receiving waterway. So when we talk about lake management, there are really four main components. I'm really going to talk about the one that's in bold up there. So the one is watershed management. You want to reduce the nutrients going into the lake. You can think of the watershed as a funnel. Any rain or water that hits that funnel goes into the bottom of the funnel, which is the lake. The second component is internal loading, and I'll touch a little bit on that. I'm really going to be focusing on these near shore areas that are really incubators for a lot of the cyanobacteria. But then also you have to get everyone who's involved in the water body, whether they visit that lake to recreate, whether they use that lake for potable water, or they live in that watershed. Getting the stakeholders involved is really critical. I am focusing this talk on Presque Bay, which in a way can be thought of as its own lake. It's not particularly deep. The average depth is about 20 feet, maximum depth about 25 feet. It's a large water body, but because it's not that deep, it can be polymictic, which means it can thermally stratify and then it can mix. And by thermally stratify, I mean if you've ever dove into a lake and it's nice and warm at the surface and you go a little deeper and all of a sudden it gets cold, it's because there's this quick change in temperature, which is also a quick change in density. So a lot of times when a lake is set up like that, it seals the bottom waters off, and I'll talk about how that affects water quality in just a second. I teach a class on watershed management at Delaware Valley University, and so I get into a lot of this stuff, how, again, rain, stormwater, produces loads of nutrients and other pollutants that then go into the receiving waterways, agricultural sources, urban areas, residential areas, goose waste, pet waste, all of it leads to this non-point source pollution that can trigger these harmful algal bloom events. When I teach watershed management, I always tell the students, if there's one slide I want you to remember from the entire class, it's this slide. It's basically the more vegetation you have, the less stormwater you have going into the receiving waterway. So when you see like a forested watershed, you can see the majority of that water is either going into the ground or up into the atmosphere as evapotranspiration. You can see only 10% of it is flowing as runoff. In a very urbanized city situation where you don't have a lot of green infrastructure, a lot of best management practices, that water can't get into the ground. That water, you don't have as much going up into the atmosphere, so that means over half of that water is runoff, and that runoff transports those nutrients into those receiving waterways. So that's my talk on watershed management. For internal loading, this is really important because when you have a situation, say here, if you look in the upper left during the summer for a deep lake, you have that layering of the water layers so that you don't have any atmospheric exchange. In that area called the hypolimnion, there's this chemical reaction that when you run out of dissolved oxygen down there, there's a chemical bond between iron and phosphorus that's broken. That phosphorus is then available for algae growth, and it accumulates. That's called internal loading. So that's something that can be really problematic in a dimictic lake. Dimictic meaning it mixes twice a year, mixes in the fall, then you get an ice over, and then it will mix in the spring, and then set up that thermal stratification. Water bodies like Presque are more polymictic, meaning it can thermally stratify, but then you can get a storm or a wind event and it can easily mix that system, so it can pulse these nutrients up. On top of that, many of your cyanobacteria have what we're called gas vacuoles or vesicles. What that means is when water's nice and still, they can float to the surface with these gas vacuoles, and that's what you see over here, that they make that surface scum. The advantage to that is they can outcompete other algae for sunlight. Then if they're starving for nutrients, they can collapse those gas vacuoles, sink to those nutrient-rich waters, and fuel up. On top of that, cyanobacteria tend to have their highest growth rates at high temperatures. So they prefer warmer conditions. That's why they tend to bloom most in the summer and late fall. And I do a lot of work on Lake Apacon, which is the largest lake in New Jersey. I started working on Lake Apacon in like the early 90s, so around 92. Excuse me. But here you can see surface water temperatures at the Mid-Lake Sampling Station. This is just July surface water temperatures, and we've had a statistically significant increase in temperature at the lake. That favors the cyanobacteria. It also is problematic for the carryover trout habitat that is struggling to hold on at Lake Apacon. Another thing I want to mention about something like Presque that's relatively shallow, you have more of an interaction of the bacteria and the organisms in the sediment-water interface. In a very deep lake, it's a very chemically driven physical reaction that iron and phosphate is broken, and then the cyanobacteria can take advantage of it. In shallower areas, these shallow areas can be habitat for the cyanobacteria, particularly over the winter, where they overwinter, but you also have a lot of exchange of nutrients between the decomposers and the photosynthesizers, so you have a lot of biology and chemistry going on at that sediment-water interface. So when you talk about shallow nearshore management, and that's what this presentation is really focusing on, we're really trying to get a handle on what's going on at that interface. And the thing that we've been seeing more over the last few decades is more and more of the overwintering cyanobacteria. I'm going to date myself, but usually in the 90s when we'd hit fall, all the lakes would turn brown. It's because all the cyanobacteria would die off and you'd get things like diatoms and chrysophytes bloom and then make the water look brown. Now we see the cyanobacteria going well into October, November, December. Sometimes we even see green ice. And one of the reasons is these warmer water temperatures that allow them to overwinter either as just vegetated cells or as aconites. These are resting spores, very resilient against all sorts of environmental changes. So they settle, temperatures go down, but then once temperatures start increasing in the spring and you have mixing, those aconites come up, they hatch, and produce more of these halves. And what's really distressing is not only do we see the surface water temperatures increasing, but again, this is Lake Apacom. This is at a shallow sampling station only 2 meters deep, but we're also seeing at that sediment water interface temperatures increasing. So now we have warmer water temperatures over the winter and the summer that allow the cyanobacteria to grow. And that area there is really, so this is Lake Apacom. That area you see over on the on your left, that's Crescent Cove. I'll talk a little more into that. But this is that littoral zone, that shallow water interface that can allow those cyanobacteria to bloom. And it not only produces those surface scum formers, it can also make the stuff that looks like horsehair, or I call it green cotton candy. It's the filamentous stuff that grows. So how are we going to manage these problematic algae? And a few years ago, I went to a conference and I saw Army Corps give a really good presentation of this methodology they developed, grabbing sediment samples and then lake water, filter the lake water, and then you're basically evaluating, you're figuring out what's going to come out of those sediments. So you can predict, where the cyanobacteria are going to show up and what type are going to occur. So we did our first experiment with this with Smith Mountain Lake in Virginia. That was our first incubator. We actually have a larger incubator now, but back in early 2024, we ran some samples of sediment and water from Smith Mountain Lake. I don't have time to get into it, but the one thing I want to point out that we found that was really telling were samples that had a lot of sand. You think of like a beach area, weren't very conducive for overwintering cyanobacteria, which makes sense. If it's sand, there's not a lot of nutrients, not a lot of organic matter. In contrast, sites that had silty sediment or a lot of organic material were great incubators for cyanobacteria. So this has allowed us to develop management strategies for specific types of areas. So if you think of beach areas here at Presque Isle, so beach area, you have a high contact between people and the water, but you have a sandy bottom, so it's not very conducive for overwintering algae. So that means if there's a HAB event at a beach area, it's probably coming in from some other source. It's probably being blown into the area. So some of the strategies we think of for a beach area for nearshore management can involve circulation, floating booms, floating wetland islands. Air curtains are very effective of preventing the cyanobacteria from accumulating along those shorelines. Maybe some nutrient inactivation. And if you're going to do any sort of algicide treatment to kill the algae, it'd be more reactionary in nature. So these are some of the strategies we've developed for these beach plans. Now, if you're at say a boat launch or a marina, here you still have contact, but it's not as direct. But a lot of times the sediments are more organic or silty in nature. So indeed the algae may be originating from these areas. So here we're looking at strategies that are focusing more on nutrient inactivation, minimizing the availability of nutrients, and more appropriately, maybe some dredging to not only get rid of the organic sediments, but also get rid of those overwintering algae. And so a lot of times, in a lot of instances, we'll use biochar as a way of removing some of these nutrients. But an application of an algicide may be more proactive in nature, and I'll touch on that as I get toward wrapping up my presentation. Then the third area are more natural areas. You think of living shorelines. These are areas where you want to promote the establishment of submerged and emergent vegetation. Having a nice vegetated aquatic wetland buffer helps to remove nutrients that otherwise stimulate harmful algal blooms. But also this is great habitat for zooplankton, for a variety of fish, and for aquatic life. So these are areas you want to preserve and protect, not only to prevent HABs from occurring, but also to favor some of these desirable organisms. So here, we're looking at, the establishment of living shorelines, establishment of submerged and emergent vegetation, maybe some fishery management, installation of fish structures, and really preserving and protect some of the young of the year fish, some of the more popular ones, like yellow perch that are found in Presque, as well as some of the other desirable species such as white perch. And this idea is if you have a shallow area where you have more aquatic vegetation, the water will be clearer. When that sunlight hits the sediments, it's going to stimulate plant growth. You'd much rather that than a HAB event where biodiversity is lower, and now you may have to contend with these cyanotoxins. So I do want to wrap up by showing you, so we've just finished a whole series of treatments in these nearshore areas at Lake Apacon. We modified Army Corps' methodology for the incubation, but I do want to talk about one proactive treatment we did at Crescent Cove. So there aren't too many algicides out there that you can really use. You're either using copper-based algicides or oxidizers. And usually what happens is you wait till you have a bloom and then you hit the algae and you kill it. Well, what we did with one of these experiments in Crescent Cove, we said, well, instead of waiting for the bloom to hit, why don't we be more proactive? We know there's overwintering algae over the sediments in this shallow area, and we know Crescent Cove is really where the HABs start in the lake and then they spread out. So instead of waiting for the HAB in August and hitting it, we went in April and we applied the oxidizer, PAC 27, immediately over the sediments. We actually got some very positive results, and we're in the middle of getting our data together, but separate from us, there are a group of volunteers on Lake Apacon that use a phycocyanin meter to directly measure cyanobacteria. Phycocyanin is a pigment that is almost exclusively produced by cyanobacteria. And the cool thing about these meters is people can be easily trained to calibrate and run the meters. So the volunteers from the Musconetcon Watershed Association, they collected data in 2024 before we did the treatment, and then in 2025 after we did the treatment. And you can see from these two sampling locations now, each year at each station, sampling started in June, ended in September, and you're talking about anywhere between 15 to 20 samples. But I have bolded here the mean values. So before this proactive treatment was conducted, the average phycocyanin reading was 29 and 34. The post-treatment year, this year, 2025, the mean it's a fluorescent unit, is 11 and 15. So we had a substantially lower amount of cyanobacteria. And the nice thing about it is it persisted through the whole year. So, and again, I don't know if this is going to happen in every case or in every year, but it was nice we did one treatment in April and really kept things under control for that entire growing season. And there were a lot of aquatic plants coming up because it's a shallow water body, sunlight's hitting the sediments, and that's what we want to do. I always go to these public meetings and I tell the public, I'd rather hear people complaining about weeds than complaining about algae. Because the weeds are easier to manage. They have a mechanical weed harvesting program to remove the weeds. You can be more selective in the plants. You remove, once you have a HAB event, you're struggling to deal with it. So that was some positive results. We're going through more of our results, but I'm going to finish my presentation by saying we're going to be doing sort of a similar innovative project here in Presque Bay, we're going to evaluate a specialized clay called lamponite, which is used as a flocculant to drag the cells from the water column to the sediment. And a lot of your sediment heterotrophic bacteria can decompose cyanobacterial cells, and there are certain genera of bacteria that can actually break down microcystins. So what we're going to do is we're going to evaluate if we use this clay to drag the cells to the bottom, will we see a reduction of the cyanobacteria and of cyanotoxins at that sediment-water interface, sort of a way of using a non-pesticidal way of managing these cyanobacteria in these near shallow areas. So, you know, we're hoping, we're going through the permit process now with the state and federal agencies. We'll be evaluating this sometime next summer in some of these near-shore marina and recreational areas. So again, this is, as opposed to starting out by seeing all those surface scums and nasty green paint spilled on, this is really what we want to see in a lot of our aquatic ecosystems. So thank you very much. That's our research team in our aquatics group at Princeton Hydro, and I thank you for your time.
Questions for Dr. Fred?
So you said you attend a lot of public meetings, so this is a question more on like the response from the community. And obviously like lake weeds and stuff are bad for recreation. People have a lot of complaints about that, especially when like a smaller lake, say like Chautauqua or something. But how, like in your experience, has anything been more effective in terms of like educating the public on that type of thing? Because a lot of times they don't even understand the difference between like a harmful algal bloom that's bad versus lake weeds that are good, like preserving the lake.
Right.
You want the weeds. So
Right.
I don't know. What's your experience with that?
So in a way, a lake, either whether it's a recreational lake or a potable lake, a lot of times you already have a captive audience. So Lake Apacon is a public lake, so it's a state park, but you have a lot of people that live around the lake, and the value of their property is directly related to the value of the lake. So they're very tuned in, and they go to these public meetings. So the Lake Apacon Commission is a state agency. They meet once a month. They have public meetings. So we're always providing information during those meetings. So watershed management can be very daunting when you're educating the public. In a way, with lakes, I actually find it easier because you almost have that captive audience because they understand that. And what spurred a lot of the research at Lake Apacon was in 2019, there was a nasty lake-wide hab that hit from June to August. And if you think of this part of the United States, your high summer season is essentially Memorial Day to Labor Day. And not only did that affect the ecology and the recreational value, it hit the local economy bad. So that was a year before COVID. And people were, like, small businesses had to close because that season just was lost. So it's very easy to convey these ideas. I mean, I still get people complaining about the weeds, but I'm like, I know, but you know, we have the harvesters, we have other tools. So it's always an uphill battle, but it just seems easier with lakes than say a river system. Yes.
I feel like a lot of people drive for little boats that have on the lake. Because if you get any people basically complain like they have their boats in the water and cyanobacteria got into their boats at all, and they ruin their motors and everything.
I'm not sure I quite understand your question.
Has the cyanobacteria had any property damage? Like, will they have had the boats in the water, right? Like, you know, throughout the winter and stuff. I feel like I pulled the boats out and they took it out and there wasn't damage to it, essentially.
No, I haven't seen anything like that. I mean, more it's a concern like, and again in 2019 in New Jersey, a couple dogs died. So I've been to public meetings where I'll talk about HABs in like, say, a park setting. There's a couple lakes. And I talk about, oh, it can make people sick, and people are just like, and then I said, oh, and it can kill dogs. And all of a sudden people are like, what? It can kill a dog? All of a sudden that really, you know, people will pay attention to that. I haven't seen any sort of impacts on boats, but I know with boats, we're very concerned about invasive hitchhikers, you know, like hydrilla or, you know, zebra mussels or stuff like that. So, you know, cleaning your boat off in and out of a water body is real critical, not necessarily for algae, but more for some of those other invasive species. Yeah, I mean, I have a golden doodle, and I won't let her, if a water lake looks green, she'll eat anything. I don't let her anywhere near that water if it's green. So yeah, dogs are very, the Regional Science Consortium has done a lot of good research on, the susceptibility that dogs have to cyanotoxins.
Does, of course, the lake over here has a lot of like wave cover. Does wave cover affect the amount of cyanobacteria in the shallow water necessarily?
I mean, it can help bring it up, but the thing about cyanobacteria is they actually like hot, still water conditions. So when they really make those surface scums, it's when there's not a lot of wave action. So if you do have a lot of wave action, like a lot of the management efforts will do it for potable drinking water, is just putting a de-strat system and mix the water column. It's not going to eliminate the algae, but what it prevents those bands of cyanobacteria, because if you're a water purveyor and here's your intake, and the cyanobacteria are coming down because they want to fuel up, and all of a sudden they go into that intake, now you're dealing with cyanotoxins, problems with turbidity, filtering, you have to add more chemicals to clean the water up. So keeping it well mixed can actually make it less of a problem. Now, if you have a major HAB, the water may still look green even though it's agitated, but overall they tend to really proliferate when there's not a lot of wind and wave action.
So thank you, everyone. I think there will be room to discuss with Dr. Fred, after the session. We move to our next session. Thank you, Dr. Fred. Of course, I know a lot of people want to interact with Dr. Fred because we are talking about cyanobacteria. And I think we will continue in that dimension. The next presenter will be talking about cyanotoxins. And the next presenter is our own Dr. Sarah from the Regional Science Consortium. Sarah, you have the floor. Okay.
All right, thank you very much. Yes, we will continue talking about cyanotoxins and all the monitoring we do here. So we look at multiple cyanotoxins across, we've had up to 42 different sites in this region. I believe this year we had 35 sites that we were looking at. So we look all over the Pennsylvania coastline of Lake Erie. So some of the goals of our HAB monitoring, we want to develop a long-term monitoring program for cyanotoxins and deliver those real-time results to the public. We sample once a week, and all of those results are publicly posted so that people can see what the toxin levels are like across the shoreline. We have developed a collaboration with veterinarians to identify cyanotoxin poisoning in pets in this region because, as Fred said, it can kill dogs. And then we also create a lot of education and awareness around HABs. So we started sampling for harmful algal bloom toxins in 2014, and like I said, we collect these samples weekly from May through October. This year, I believe our first sampling was actually April 30th, so technically now it's April through October. So it's anywhere from 20 to 22 weeks, depending on funding and things like that, and we access all of our sites from the shoreline. So they're either taken in a pair of waders, walking out into the water and sampling at elbow depth, because we also collect bacteria samples at the same time, or they are taken with like a swing sampler, big stick you put in the water and you pull the end of it and it collects your sample that way for areas that you can't get in the water like marinas. So we have 27 to 35 sample sites. Like I said, this can vary every year. Sometimes we add inland lakes and things like that, but we always have our set base ones. We sample Presque Bay, the state park beaches, areas of East and West County, and then we also take samples for Erie Waterworks and Northeast Drinking Authority. And like I said, we've added Edinburgh Lake, and in the past we've also added Lake Pleasant and Lake LaBeouf. So usually we collect our samples on Thursdays. This year we had to switch that to Wednesdays. It all depends on staffing and things like that, but it's always once a week. And on Presque State Park we have 14 sites. 6 of those are beaches, and then the other 8 are along the bay. So our beaches sites, you can see, have this little umbrella up here. And these are the regular sites that we sample every day of the summer for E. coli concentrations. So these are those same sites. We just sample them once a week for HABs as well. And then for Hab specifically, we collect these 8 that are along the bay. So we get Tributary for Scott Run, Vista 3, Sturgeon Bay, Niagara Boat Launch and Ferry Slip, Marina Lake, Perry Monument, Horseshoe Pond, and Boaters Beach. Our idea is to get like the full span of the bay, especially at public access points where people are most likely to be interacting with that water. And then on the Presque Bay, like downtown side, we have 5 sites. So Erie Yacht Club, Cascade Creek, Liberty Park, Dobbins Landing, Mill Creek, Garrison Run, and South Pier. So, East and West County, we now have 5 sites in East County. Like I said, we like to keep adding sites. So we have East Avenue Boat Launch, Lake Cliff Boat Launch, Shades Beach, Freeport Beach, and 20 Mile Creek. So you see that same kind of consistency of beaches and public access points are what we are most interested in. And then on the west side, we do Walnut Creek, Avonia Beach, and Elk Creek. So, I love this table. I think it's really cool. We do 4 different cyanotoxin tests. So the first of those is a microcystins nodularis test. We see a lot of hits for microcystins in this area. Pretty much every week we have microcystin exceedances for dogs. And then we also do tests for cylindrospermopsin, anatoxin, and saxitoxin. So your hepatotoxins, your liver toxins, are your microcystin and cylindrospermopsin tests. And then your neurotoxins are anatoxin and saxitoxin. Everything is also a dermatoxin. It'll make your skin feel itchy, usually, and red. That's me. We use gold standard ELISA test kits for this. Like I said, the first one is a microcystins nodularis kit, and we also use a quick lice for that, so we lice all our cells within it takes maybe an hour to lice everything, and then you can run it all. We want our maximum possible toxin levels, so we lice all cells for these. The other 3 tests use a freeze-thaw process to lice their cells. You can't use quick lice on those. It's only approved for certain tests. That's why we have to do a freeze-thaw. And then as you can see, our sampling has varied quite a bit over the years. That's usually funding dependent. Sometimes it's staff dependent, but usually this is funding dependent. So you can see how much it has changed from 2019 up to 2025. Another thing we added to our analysis for this season is something called Bloom Optics. It's a really cool AI program where you take a little droplet of water under a microscope, and that microscope takes pictures. It's called an iolite microscope. You upload those pictures to the AI program, and within about 5 minutes, it's never taken longer than that for me, it tells you whether or not there are Hab cells in that water. It's really cool. It does identify what type of cell it is and everything. So this is an example of one where a harmful algal bloom was detected. It highlights them. I don't even know how I can tell. They're so tiny. But it highlights them and tells you what it believes it is. So microcystis in this case, and then its percent certainty. So 87% and 84%. It tries to tell you the average density, the total number of colonies, all this interesting stuff. I always add in our toxin levels that we calculate to this, so that's part of that data set. It's really cool. So we did this specifically for our sites this year that we knew had exceeding toxin levels for dogs, or a visible scum. It takes a decent amount of time to actually do each sample with the water droplet on the slide and taking the pictures, so it wasn't feasible for us to do every site every week. It basically would have been someone's full-time job to do that. So we did it specifically for our sites that were exceeding. And like I said, very quick results. It'll say ND for no Hab detected, or it'll say Hab for Hab detected. So back to our toxins, some advisory thresholds that we go by here I have highlighted. We look at the threshold for dogs, which you can see varies based on each toxin. In all my figures, I have that dog level highlighted with a bright green line across every graph, so it's easy for you guys to see. Don't feel like you have to memorize these numbers like I have. But you can see there's also a recreational public health advisory, as well as a recreational no contact advisory. We do not follow drinking water thresholds for people because you shouldn't be drinking out of Presque Bay, so we don't have to look at those. Okay, so about to hit you with a lot of figures. So for microcystin for the beaches this season, these were some of our results. So as I said, that dog level is this bright green line that's going across. And you can see how many times over the season we had advisories. So actually not too bad, but this is still usually, like Fred was saying, since beaches are so well mixed with wave action, you really shouldn't see a lot of harmful algal blooms out there, especially not at a level that is unsafe for dogs. But nothing was unsafe for people, so that's great. Nothing bad for our beaches. And Zone 5 saw the majority of our exceedances. Zone 5 is very protected. It's over here compared to every other zone. So it does make sense that area gets less wave action. You would expect to see more exceedances for dogs in that region. So here's our saxitoxin results for the beaches. You'll see that I don't believe I have a single graph for cylindrospermopsin or anatoxin because we did not see exceedances for those toxins this year. I'm only showing you guys our exceedance results. So once on this one, you can see your dog level is only 0.02. Once again, I have that highlighted with that green line. And we did have saxitoxin exceedances, that's the neurotoxin, for dogs at 3 different beaches this season. So we tend to see this trend with saxitoxin, where at the start of the year, you have a lot of saxitoxin exceedances, and at the end of the season, we have a lot of saxitoxin exceedances. And then microcystin just takes over in the middle. Once again, there was no human exceedances. For Presque Bay, this is the Presque State Park side of Presque Bay. I can't put them all in one graph. It's horrible. All right, so you can see we had a couple really high results here. This one, thankfully, is still underneath the human recreational threshold because that is actually 8 parts per billion for human recreation, but we had it all the way up to over 4.7 parts per billion. That's really high. That's really dangerous for dogs when their limit is only 0.2. You can see a lot of other dog exceedances down here for various sites. It's not, I have a map that I'll show you guys later exactly where it's popping up, but there was a lot of exceedances for microcystin. And then for saxitoxin in those same areas, there were less, but a lot of them at Horseshoe Pond. So you can see that yellow pops up a lot. And once again, Horseshoe Pond is out here. It is super secluded. There is not a lot of wave action. There's not a lot of really water moving around at all. So basically, once those habs got in there, they stayed for the whole season. They liked it. Here is the downtown side of Presque Bay. Once again, lots of dog exceedances. You see this one's a little higher, but keep in mind it's not even remotely as high as what we saw 2 slides ago. It's still under 1 part per billion. Everything's fine for people, but it's scary for dogs. Keep in mind a lot of these places a dog can't really get in the water, like Erie Yacht Club. There's not really a shoreline there, it's a marina. And then, okay, here's the downtown side. So once again, towards the end of the season. So for the East County, for microcystin, once again, looking at that, we do get more exceedances on the east side than we do on the west side. So you can see some of these. Once again, it's really not that high. All safe for humans, but not looking great for dogs. And there's your saxitoxin results. And then our west side, typically, in like the past 2 to 3 years, the only site that has exceeded for dogs has been Avonia Beach. But this year, Elk Creek wanted to be involved. So we have a tiny dog exceedance at Elk Creek here, but still that is over the dog limit. But otherwise it was all Avonia Beach again. Walnut Creek is just staying out of it. And then our 1 exceedance for saxitoxin at Avonia Beach. So this is what I like to show because I think this makes a lot more sense than looking at graphs. It's showing you guys where all of those exceedances have popped up over the summer. These are all dog exceedances, nothing human. But if it's outlined in yellow, it's for saxitoxin. So this is our first sampling of the year at the end of April. You can see there was no microcystin exceedances, but for dogs it wasn't safe at the Ferry Slip, Horseshoe Pond, or Zone 3, which is Mill Road Beach. And then our next sampling was May 21st, and you can see that all our saxitoxin has disappeared, but microcystin popped up in Marina Lake. And then it starts to spread across the bay. So I won't go over each site, but I want you guys to just kind of watch how those signs are popping up. Concentrates up there near the channel. Spreads across the whole bay again and a little bit outside the channel. So as you go from week to week, you see how it can move around. We have saxitoxin pop up again at the start of July. And then here you'll start to see we had to stop sampling saxitoxin every week. So saxitoxin's here. That exceedance remains in place to the public. We leave that up as an exceedance until we test again. So we weren't able to test again till the week after next for this one. So it'll remain there even though it wasn't tested. You can see microcystins all over the bay. It also popped up over here in East County. And we have it popping up in East and West. So both those appear. Becoming very widespread as we're heading later into July. Saxitoxin was retested. It's still at Horseshoe Pond. And then saxitoxin just kind of went everywhere. So microcystin was only at 4 sites, and saxitoxin popped up literally kind of everywhere. All over East County, every site. And then at a beach, at Beach 2, and then all over the inside of the bay, and also at Avonia. Very interesting week there. And like I said, all those stay because we weren't able to sample again the week after that for saxitoxin. So microcystin was tested again. A couple more sites, a couple different sites, but still. And then when we tested for saxitoxin again, some of them went away, but a lot of them were still there. No microcystin exceedances this week. And saxitoxin wasn't tested. So these are all still from the previous week. But microcystin went away. You can see how that changes again. We're starting to slow down a little bit. And then they all pop back up. This was our last sampling of this year. We have not had a year where we ended with no exceedances in a very long time. So every year we have to stop sampling before we actually see exceedances go away. So just looking at our number of advisories over recent years, I'm specifically going to focus on 2025. We had 158 dog exceedances for microcystin and 39 dog exceedances for saxitoxin. Keep in mind that is a neurotoxin. So that's what can really kill a dog very, very quickly. And once again, I wanted to keep in mind that these are variables, so you have to keep, remember that each year, sometimes we were able to test once a week for saxitoxin, sometimes we weren't. So just please keep that in mind when you look at the numbers. So like I said, we post all of this as soon as we can. I finish the results, I email them out to our partners, and then I immediately go online and update this map. So the public know basically as soon as everyone else. This is on our website. It's also on the health department's website. I know you can't click on that link, but it's easy to find on our website. You just go to our website, click on the harmful algal blooms, and there's a local HAB map right there. So every logo with a little no dog means that there's a dog exceedance at that site. If it is green, it means it was safe. So this is what we left off on at the end of the season. This is from October 2nd. So like I said, we email those results to our property managers and they post signage. These signs right here. We do have different signs for animal and human levels. We only had 1 human exceedance this season, but it also exceeded the limit of the test. That was at Eaton Reservoir, so it wasn't one of the sites on the map there. And when we retested it, it was below that human level. We had to dilute and retest. That's another unfortunate thing, is that the human level is outside of the limits of an ELISA test. So if you don't have a visible scum, you don't know to dilute it ahead of time, sometimes you can have results that shoot off really high and you have to wait till next week to retest them to see what they really are. So to be cautious, we always say, human recreation, and then the following week we're like, okay, no, it's all right. It's just a dog recreation. On these signs, there is a QR code to the Erie County Health Department's webpage for that HAB map. We also do, like I said, education. We have a big HAB lab. We actually just used this last week as part of our forensic science escape room. So this is a really cool tool that we have. It has a lot of dogs in it because, like Fred said, when people say it can make you sick, they're like, okay, I can get sick. But when you say it'll make your dog sick, they're like, what can we do? How do we solve this problem? It really motivates people when you bring their dogs into it. Plus, dogs are so much more sensitive. Some next steps, we want to continue our weekly monitoring. We want to keep doing this every year, preferably for all 4 toxins as frequently as possible. We are working on creating a predictive model, and then we want to do some comparisons, including to our Bloom Optics results. I have a intern who is super interested in that, and he wants to be able to compare those to our current results, so maybe he'll do that next summer for us. And then this project was made possible through PA-DEP Coastal Resources Management Program and Great Lakes Observing System. Thank you.
We just take one question.
Is it just that dogs are more likely to consume water with the HABs in it, or is there a physiological reason that dogs are more sensitive to the toxins?
It is, I mean, both things definitely feed into it. Dogs are way more likely to consume it, but they're also just smaller and more sensitive. So it's kind of both. Yeah.
Thank you, Dr. So I know we have many people that we have questions, and during the lunchtime, there will be room for questions. Please find any of our presenters and ask them your question. And we go to lunch break now. We have 1 hour lunch break. And after that, we move to another session where we have Dr. Oli Best that will be sharing us during that time.
So if you were a presenter or a judge or one of our high school students, there's going to be lunch in room 108 over there. And if you guys want to stick around, hang out in there, there's tables maybe out there in here. You can stick around and ask questions or anything like that.
Yeah. Yeah. the field manager here for the RSC. We're going to go ahead and just get right into it with our first presentation with Brian Winston from the Watershed Conservation Research Center at Allegheny.
Thank you. Is there a clicker? There it is. Sorry. All right, everyone. Thank you for coming back after lunch. I know the pressure's really on me. I gotta do a presentation, and I have to keep everyone awake after they ate lunch. So, hopefully you enjoy it. As he said, my name is Brian Winston, and I am part of the Watershed Conservation Research Center. For anyone unfamiliar with us, we are located at Allegheny College. We're made up of 4 full-time staff. That is professors and founders, Dr. Casey Bradshaw Wilson, Dr. Kelly Pierce, research scientist Mark Kirk, and myself. Today, I will be presenting the results from a long-term evaluation of 3 stream restoration projects in the French Creek watershed. So the primary area that the WCRC operates in is the French Creek watershed. French Creek has its headwaters up in western New York, and it feeds into the Allegheny River just south of Franklin, Pennsylvania. French Creek is renowned for its high biodiversity, and it was the 2022 winner of the Pennsylvania River of the Year. In French Creek, you'll find 27 different species of freshwater mussels, 4 of which are considered federally endangered. There's also nearly 100 different native species of fish found, including the environmentally sensitive brook trout and 15 different species of darters. Within the watershed, you'll find about a dozen different species of salamanders, including the renowned eastern hellbender, and then river otters, black bears, and beavers are among the mammalian species found in the watershed. So despite its relative health, and its high biodiversity, there are a number of anthropogenic stressors that face the watershed. Among these are erosion, turbidity, invasive species, algal blooms, and runoff. And pretty much all of these stressors are the result of poor land use management within the watershed that will lead to a degradation in water quality. So in order to improve stream habitats, the WCRC has made it a priority to partner with local conservation partners and private landowners to complete 3 in-stream habitat improvements on Woodcock Creek, which is a tributary to French Creek. The main goals of these projects were to reduce sediment in the water by reducing erosion and stabilizing stream banks, and to improve habitat for aquatic organisms. So sedimentation has been and continues to be one of the largest stressors of stream environments across the country. And to better determine the efficacy of these restoration efforts, the WCRC has performed biotic monitoring pre and post restoration at each one of our sites. And then during the summer and the fall, we partnered with the RSC actually to monitor nitrate, phosphate, E. coli, and enterococci levels in different areas of the streams to determine how our restorations have impacted the local water quality. And finally, these projects have provided a great on-site educational opportunity for Allegheny College students. So we currently have 3 sites where we've performed our restorations. You can sort of see them up there, highlighted in different areas. I mentioned, though, that these sites are all located in Woodcock Creek, or the Woodcock Creek watershed, which is a sub-watershed to French Creek. So for reference, French Creek, the main stem, which goes there, that runs for about 117 miles, and the watershed it's located in is about 1,200 square miles. Well, the main stem of Woodcock Creek runs for about 20 miles, and it has a watershed area of about 50 square miles. So Woodcock Creek is primarily forested but there is a mix of agricultural and residential developments as well. And the 3 sites where we have our restorations are Woodcock Creek at Craig Road, German Run at the Telly Ho property, and Woodcock Creek at Stainbrook Park. So when designing these different restorations, each site that we go to presents a different set of challenges. So these plans for the restorations are primarily done by the Pennsylvania Fish and Boat Commission. And when they're planning them, they have to use a number of different restorative techniques to address these different challenges. So some of the common structures that they will use in the restorations are log vein deflectors, root wad deflectors, and modified mud sill cribbing. And these different techniques work in unison to help stabilize stream banks, to trap sediment, and to reduce and redirect high flows. So our first site, Woodcock Creek at Craig Road, this restoration took place in August of 2022, and the main goal of this project was to reduce stream bank erosion, which was occurring in those areas, and to also reduce the downstream transport of materials that were causing sedimentation as well. So in cooperation with the local landowners, the Pennsylvania Fish and Boat Commission, and the Crawford County Conservation District, we installed log vein deflectors there. You can see that where they are in relation to the stream bank. Behind that, we put in modified mud sill cribbing. Then we also added root wad deflectors further up the stream. So you can see what the site looks like now, 3 years post-restoration. So that's the same bank as what you see there. You can see that the stream bank has been stabilized and that further erosion has been avoided at this area. Our second site was located on a small unnamed tributary to Woodcock Creek that we would colloquially refer to as German Run at the Telly Ho property. So due to this one took place in June of 2023. Due to recent land use changes upstream of this site, there were lots of high flow events that we were seeing, particularly during storms, which were causing, you could just see there, severe erosion and once again, downstream sedimentation, downstream deposition of large amounts of material at this site. So we once again partnered with our local conservation groups and with the private landowners. And during this restoration, log vein deflectors were installed along the stream banks where that erosion was occurring. And large woody debris was also added in the actual stream to help to slow down some of those high flows and to also trap some of the sediment and material that was being transported downstream. And another additional benefit of these different structures is that they help to increase the number of deeper pool and riffle habitats in the stream, which is great for improving the habitat heterogeneity as well. So our most recent restoration project took place at Woodcock Creek at Stainbrook Park in June of 2024. If anyone's familiar with Stainbrook Park, it is located just downstream of the Woodcock Creek Lake Reservoir and flood control dam. So during storms, as water was released from the dam, it was rushing through this area, creating really bad erosion, also creating downstream sedimentation again. And then in addition to that, this area of Woodcock Creek was primarily just one big homogeneous run with very little habitat for diverse fish and macroinvertebrate populations. So at this site, once again, you'll see a theme. We added log vein deflectors along the shoreline. We also added large boulders to the stream. You can see there on the left, your left, yeah, the log vein deflectors being installed. You can't see them as well, but there is large boulders all out throughout here as well. You can see there, stream bank has been stabilized here as well, and those additions again help to add in more deep pools, more riffle habitat. And then in addition to that, in the spring of 2025, it's a little bit hard to see, but if you can see the little white tubes there in the background, Allegheny College students came out and they planted over 100 native trees and shrubs in the area, which will help to stabilize the riparian zone and also provide habitat for different terrestrial animals at this site. So, beyond just performing the restoration, a lot of times you might think of it and think, oh, we're done, we did it, everything will be fixed now. But no, that's just the start of it. Equally important to performing the restoration is being able to evaluate their success. So in order to do that, the WCRC has performed extensive monitoring pre- and post-restoration at each one of our sites. So prior to the restoration, we go out to the site, we do seasonal electrofishing surveys, we collect aquatic macroinvertebrates, we evaluate the riparian habitat at each site, and we analyze water chemistry and hydrology. So after the restoration is actually performed, we come back out. Every year we repeat those surveys. In addition, we will collect bi-weekly water samples to get an idea of what the water quality is at each one of our sites. Then, more recently, we've actually started using some temperature and light loggers that we can keep out there most of the year to be able to track the temperature and turbidity of our streams. Today, I will be focusing on the results from our fish surveys and our macroinvertebrate surveys. All right, so here we have the results from our fish surveys at our first site, that was Woodcock Creek at Craig Road. So looking at the pies, each slice of the pie represents the relative abundance of different families of fish. So looking at these two, it doesn't seem like relative abundances have changed all that much, but you can note a few differences that are notable. So one of them is the addition or the near doubling of the relative abundance of the family of fish called the Catastomidae, which are our suckers. And you will also note that there is an addition of a new family of fish, which is Amidae, or the bowfin, that we found at this site post-restoration. So in addition to looking at those fish populations, which might be what comes to mind when you think of these restorations, we also, though, monitor macroinvertebrate populations, as they're a very important indicator of ecosystem health. So in particular, macroinvertebrates belonging to the orders of Ephemeroptera, Plecoptera, and Trichoptera are very useful as clean water indicators. So the Pennsylvania Department of Environmental Protection has established a standardized methodology that we follow, that we will we'll assign each macroinvertebrate sample a benthic index of biotic integrity, or a BIBI score. So these BIBI scores range, you see the chart only goes to 50, but the BIBI scores range from 0 to 100, with 100 being the highest possible indicator of biotic integrity, and scores below 50 represent impaired streams, or scores that are impaired. So looking at the average BIBI score, this dashed line there, that's when the restoration occurred. So you see not much change overall in the average BIBI score pre and post restoration. The one thing you can note though, this sharp decrease in BIBI score immediately following the restoration, followed by this positive trend over time afterwards. And now looking at the results from our electrofishing surveys at our second site, German Run at Telly Ho property, you begin to see a little bit more of a change in the fish family relative abundances. So a big one you want to take note of is the increase in this family. That is the Persidae family, which are our perch and darter species, increasing from about 8% up to almost 12%. And then also a large increase in the Cotidae, or the sculpin family relative abundance, from under 3% to almost 12%. Looking at the macroinvertebrate results from our Telly Ho property surveys, you'll see sort of a similar thing. Not much of a change in average BIBI score. There is a slight increase post-restoration. But note that similar to our first site, we see this decrease shortly after the restoration, followed by this positive trend increases in the BIBI scores over time. Finally, our most recent site, the Stainbrook Park, Woodcock Creek at Stainbrook Park site, you'll see the largest difference in the relative abundance of our different fish families. Take notice though especially, another large increase in the Percidae or perch and darter families. And then also the addition of 2 new families that we've seen post-restoration, the Cotidae, the sculpins, and then the Associdae, which is our pike and muskie. Here, you'll see, once again, just a slight increase in Bibby score following the restoration. You don't see the major drop-off, although scores were pretty low before and just after the restoration. And then you are seeing a slight positive trend. With this just being a year ago, though, due to that recency, as we get more data points at this site, we'll hope to see even more of an increase in the Bibby scores at this site. So, we are now 3 years post-restoration at our first site, which was Woodcock Creek at Craig Road. Although we did not see a major change in the family relative abundances at this site, we have seen an increase in fish species detected from 31 pre-restoration to 40 post-restoration. A big part of this has been an increase in darter diversity, namely an increase in log perch, which is pictured there, and then variegated darter populations. So the good thing about seeing an increase in the darter population is that they're an environmentally sensitive group of species, which would indicate a positive sign as far as water quality and habitat availability at this site post-restoration. Then we've also seen an increase in the sucker relative abundance, which I mentioned. Another good sign, because suckers are a highly mobile group of species, so that's a good sign that we have good stream connectivity at this site. And then finally, that there was no major change in macroinvertebrate community at any of the sites, really. But this isn't all that unexpected. As previous studies have shown, there will usually be a lag time in the recovery of a macroinvertebrate community at different restoration sites. Because when you go in, you do this restoration, you're really impacting the current population, which will lead to that immediate drop in the score. But then usually there'll be a recovery over time, and some of the more sensitive taxa will be able to recolonize the sites. At our second site, German Run at the Telly Ho property, we're now 2 years post-restoration. This site, like I said, not a major change in relative abundances, but we have seen an increase in the number of species detected from 11 pre-restoration to 20 post-restoration, highlighted by an increase in darter diversity from only 1 species pre-restoration to 4 species post-restoration. And that's our rainbow darter right there. Also, we have seen that large increase in sculpin relative abundance, which isn't super surprising because sculpins will typically inhabit the shallow, fast-moving riffle habitats, which I mentioned are a little bit more common at this site following the restoration. And like I said, this one had that slight increase in the sensitive macroinvertebrate taxa, which we saw the decrease immediately following restoration, improving over time. Now finally, our most recent site, Woodcock Creek at Stainbrook Park. We are only, like I said, 1 year post-restoration. So we have seen actually the same number of species detected pre and post-restoration at 33 species. One of the reasons for this may be that there's high species turnover at this location, just with its proximity to the reservoir and the dam. Species will come and go pretty quickly from this site, whether it's up into the reservoir or get washed out during storm events. Despite seeing the same number of species, we have, though, seen an increase in the number of game fish and larger individuals at this site, including the muskie and largemouth bass. And this was another one where we did see that slight increase in sensitive macrotaxa, but hopefully over time we'll see even more of a change to the positive. So because streams are constantly changing and new threats continue to emerge, we are committed to performing ongoing restoration efforts within our local watersheds. We currently are planning 2 chop and drop restorations, one of them being on German Run, one in the Oil Creek watershed. That involves cutting down trees in the riparian zone, dropping them into the streams, which will trap sediment that's coming downstream, and will also create structure and slow down flows. We also have projects we're planning to improve fish population connectivity, including the removal of old dams and the replacement of old outdated circular culverts with newer, less restrictive elliptical culverts. So I'd like to thank many of the groups that have funded our research and our restorations, as well as different conservation groups we've worked with. Finally, any questions? Yes?
So, just before you finish, you talked about replacing culverts from circular to elliptical
Yes.
section. What would be the advantage of changing the shape?
So, with these circular culverts, a lot of times the way that they're very narrow. So when water is coming through the circular culverts, it is shooting out of them, which basically creates this shotgun blast coming out of the culvert, which creates these really large, they're called sluice pools. A lot of times it'll result in this, which is a head drop. And when you see drops like this, most of our species have no way of getting up into that culvert. So if you have a population above the culvert and below the culvert, they just get completely cut off. One thing happens in one of those areas, you can lose an entire species from a certain area. So these newer ones are designed with the idea that the stream is not narrowed going through the culvert, so that way, you know, they're able to move up and through those culverts. And also, generally they try to leave some dry passage so terrestrial animals can travel through the culverts as well. Any other questions? Yeah.
Why were those particular restoration sites selected? What's the rest of the watershed look like?
Yeah, so one of the big things, like when we were first starting out, was just having landowners that were interested in having us come out and do restorations. So a big part of that is areas that have really bad erosion and the landowners are dealing with the loss of their private property. So you kind of get invited out to do that. And then, I mean, a lot of the decision making comes from Pennsylvania Fish and Boat as well, as they're the ones that kind of spearhead the actual process of the restoration. And then we come in on the back end and do a lot of the monitoring. But yeah, and then all three of those sites just were very impaired. But there are also other impaired areas which we would love to come out and do a restoration, but either we're not invited onto the private landowner's property or we just don't have the funds for it. So it's sort of a balancing act in what we're able to do. Yes?
So I noticed as well as a deep post-restoration that you mentioned. So was that as a result of the disturbance due to restoration, or are there other factors contributing to the deep that you see?
So, like this? Yeah, so you can see that's all like 1 and 2 months after the restoration. So this one was kind of low before as well, so you might see it better on that one. But yeah, it's that these sensitive species would get disturbed during the restoration project. So you get a lot of sediment in the water, you get a lot of the actual structure on the bottom gets disturbed. So these animals will either die or get washed out. But then, like I said, over time, they do have that ability to recolonize the area.
All right, we are just about out of time for questions. So
Thank you.
Brian. All right. Next up, we have Matt Avellini, Kenneth Alford, and Ridge Gray from the University of Pittsburgh at Bradford. This didn't open properly, did it? Is this it right here?
No, it's not. You sent it to the email, Sarah.
Oh, you didn't load it when you guys came in? Sarah, it's in your email.
You sent it to your email? This one too.
There was only one presentation on it.
Could you just quickly check real quick? I think it should be there.
Yeah. Not that I know of, but it could be a possibility.
Oh, yeah, you know what point you have?
Where exactly?
Yeah, see how quickly you guys can memorize it. Oh, it did not fill.
Yeah, we haven't seen that in studies locally. So difficult trying to get the water to
Yo.
Hello, everybody. Sorry about that little confusion. Good afternoon. My name is Kenneth Alford. I'm here with
Ridge Gray and Monovan.
and today we are here to present our research on the water quality and microbial activity in selected natural springs in the McKean County area. So let's talk about why we decided to do this, the importance, stuff like that.
So a little background on our project. So about 12% of PA residents consume natural spring water, either primarily or secondarily. So especially in the western side of PA here, where it's more rural and spaced out, especially in McKean County as well, we have decided to look at commonly used natural spring water locations, especially because residents do consider them pretty much clean because it looks clean and it tastes good. So we wanted to look at the actual chemical properties and microbial properties to see if it truly is that way, because studies have been done and shown that 91% of roadside springs have failed at least one drinking water standard set by the EPA. This also brings us to our research aim and objective. So our overall aim, again, is to assess the drinkability and safety of natural spring water sources in McKean County. So we looked at 3 different areas, which is Lewis Run, Fosterbrook, and Derrick City, which are about 30 to 45 minutes from the University of Pittsburgh-Bradford campus. We then brought those samples back, and then we first did water chemistry analysis, as you can see up there. We looked at key parameters like pH, conductivity, dissolved oxygen, iron, total dissolved solids, or TDS, and chlorine. After this, we then collected more samples and then streaked them on nutrient agar culture plates to then incubate for some days to see if any microbial or bacteria colonies would grow, look at the morphology of that bacteria and grain stand to see if we could see them on a microscope. And then after that, we collected all the data and results and compared them to EPA drinking standards.
So these were the locations we used. The first one was the Lewis Run spring. That one was off a highway on the side of the road. It was covered by greenery and all types of forestry. We tried to look and see if there was any visible bacteria or anything you can notice in the area. It looked like it was connected through pipes that ran underneath the road on the area. And as soon as we got there, we just took samples. Every location we got there, immediately took the samples with sterilized water bottles, and then we tested them with a multi-parameter. And then the next spot we visited was the Fosterbrook spot on this side. This one was in a rather the state of the area wasn't as fine as this one. This one had the grading was kind of ate at maybe from a bus. When we were testing the water, I even fell in that little ditch and got kind of soaked. There was the pipes themselves connected uphill and we couldn't like figure out where they kind of like led up to, but they were like going all the way up to this like mountain rest area that was near us. And then the last spot we visited was the Derrick City Spring, which was connected to this big up like plate, as you can see, that kind of like collected all the water, maybe for people like, we didn't understand what it was made for, but I just assumed it was for people to like just come and scoop up their whatever they wanted to grab. And yeah, so as we said, we kind of tested the parameters using a parameter. We tested for pH, chlorine, iron conductivity, dissolved oxygen, and TDS. pH is to measure the acidity or alkalinity, and extreme levels could lead to unsafe water, corrosiveness, things of that nature. Conductivity is reflects like dissolved ions and minerals. Elevated levels can show risk of runoff contamination. Dissolved oxygen is it indicates how healthy and oxygen-rich the area is. If low, it may lead to organic pollution. What else? Iron, high concentrations of that can affect the taste and color of the water, and also indicate corrosion or mineral leaching. TDS, total dissolved solids, that shows the concentration of dissolved minerals in the water. High values of that can also signal contamination. And chlorine, none of the samples we found, none of the treated water had any chlorine in it. So, you know, it just shows that also all the sources we had were untreated. But result-wise, most of the results were in the safety ranges. Only one that was kind of iffy was Lewis Run. That one had a pH of below 6.5, and that tells us that the water is okay, like overall, but it doesn't necessarily mean like meet all the it doesn't mean it's like biologically safe. And then after that, we decided we examined the microbiology quality of the water and some samples to see if they contain any bacterial activity.
So what our microbiological analysis showed, as you can see up there, Fosterbrook and Lewis Run all had bacterial colony growths, especially heavily in Fosterbrook, and then the very top there in Lewis Run. Derrick City, on the other hand, did not have any bacteria growth, or very little. This helps showcase two different things. Number one, obviously the visibility that even though the water looked clean and probably potentially tasted good, that there's still stuff that we can't see in the water, and we can now visibly see that through Fosterbrook and Lewis Run. But even then, because we use nutrient agar plates, it is non-selective, so that doesn't mean all the bacteria will be shown, so contamination cannot fully be ruled out as well. So the visual clarity, like in Derrick City, does not mean it's safe, which is why we further tested it onto the next one, where we then identified the bacteria themselves. So in the chart on the left side, we have the different types of bacteria that we looked for, and then what they specifically show. Majority of them are naturally found in plants, like plant runoffs in the soil. So while you don't want to ingest them, they're not too harmful, but the two main ones we wanted to look at were obviously E. coli and Aeromonas, as they are very harmful if ingested by a human or any animal in the area. So as we can see, Derrick City again kind of cleared the board and showcased that it's pretty clean. Fosterbrook did have a whole bunch of bacteria, but it skipped out on the two very harmful. But Lewis Run continued to show the most problematic, as it had both E. coli and Aeromonas. And then in the bottom left, you can see Lewis Run and Fosterbrook, the gram stain through the micro that we saw. And just overall, just the presence of this diverse bacteria indicates that there is environmental contamination, even though it's not visibly seen with our own eyes, and that this unregulated and untreated natural spring water, which is commonly used by the residents, needs to properly be treated.
Yes, just discussing the results we had. So a lot of it, just you can see patterns in it. Some of the sites do have contaminants, and some of the sites don't meet some of the standards we have set or the EPA has set for drinking water. But really what we're trying to bring out here is all the spring water people drink and consume is not regulated, and it needs more regulation and testing. And people shouldn't drink water that has these possible contaminants. Thereby, we just want to highlight information and data we've collected regarding these sites. And yeah, just to conclude it, none of these sites really just met the standards we had. And just because it looks clean or tastes clean, or even tastes better than bottled water per se, people shouldn't be drinking it just due to the possibility of contaminants. And we recommend that you always filter or disinfect the water you collect, or just stick with using tap water that's been regulated. And just we also need to implement regular testing for people who want to drink from spring water sites. Thereby, we just had to highlight the need for regular testing as well. And we're just acknowledging that this was funded by undergraduate research fund at the University of Pittsburgh or Bradford with the help of some of our professors there. Thank you.
Any questions? Yep.
What process did you guys use to identify the different species of bacteria?
We didn't identify any of them specifically. These are the possible bacteria. So we plated them on just nutrient agar, and then we gram stained them to find possible bacteria based on their morphology. So we have not technically identified any of them. Thereby, it also requires further analysis and further testing. Yep.
What volume of water did you put on your plates?
On the plates, it was a small volume. I will have to get back to you about that. I'm not too sure exactly the volume that was used. Of course.
I think it was Lewis Run, but the one that had the that was acidic, that had the low pH.
Mm-hmm.
What do you suspect might have caused that to be different from the other ones?
It could be just the pipe that has been set up by people in the past that could have something leaching into it, or just an environmental contaminant leaching into the pipes. And that lower pH can also signify that there's possibly a higher concentration of metals in that, just because the metals are leaching into the water now due to the lower pH. So I'm not too sure exactly what's causing the low pH, but because of the low pH, there's also higher TDS pretty much.
To go off of that, it was also literally directly on the side of a highway where you have lots of passing cars, and it's just wide open and just dripping down. So it very well could just be runoff from the road and stuff getting kicked up, and just the chemicals, especially from car pollutants itself, that just cause it to just become a little more acidic because it's just wide open. The other two were kind of on these back roads, so it didn't really have that ability. Even though Fosterbrook, it came down from a hill, so there's a lot of potential for a lot of runoff there. But again, it would be more natural from like plants and stuff, which is why we saw a little more of the Fosterbrook like potential bacteria that we see in soil and plant runoff compared to just outside pollutants like Lewis Run.
Of course.
Be honest, who drank the water?
I'll be honest though, while we were there, we had like 5 cars show up and fill water bottles or like large containers of water.
Yeah.
So it's actually a lot of people who consume this, and then one of the sites had like continuous people. Like we were there for like 30 minutes, people just kept coming and going.
Yeah, on the Derrick City one, which thankfully was kind of the healthiest one, we had 2 guys come up to us on motorbikes.
Yeah.
And they literally had these big jars of just empty containers that they dug in there and filled up. So thankfully it's a little more safer than the other 2, otherwise they would have some problems. But they said that people constantly go to it. And there's a house, so it was like right next to a house in an open field. So like people constantly come.
Derrick City is the one with the big round like thing.
Yeah.
Where people were like scooping out.
They came up and they just went right in there and scooped it out. And it was pretty much filled, the plate itself on the bottom was completely filled with a lot of algae. There wasn't any, like, we didn't see any organisms in there, but it was a lot of plant leaf litter and just a lot of algae. So they just went right in.
So the EPA has been trying to close down these spring water sites, but unfortunately the locals prefer to have them. They believe it to be untreated, perfectly pure and natural. So it's really a battle between the locals and the EPA trying to close them down and locals actually wanting them. But I'd say with proper technique, heating your water, treating it properly, you could consume it still. So it's just a matter of kind of putting out the awareness, telling people that, hey, your water is not perfectly safe, but with these additional steps that you can take, you can make it a little bit safer for yourself and the people around you. Any other questions? Perfect, thank you.
All right. Let's see here. Next up, we have Andrew Samuels from the Environmental Science and Engineering program at Gannon University.
All right, well, good afternoon, everyone. My name is Andrew Samuels, and I am a master's student at Gannon University, pursuing my degree in environmental science, and today I will be presenting to you my graduate thesis project, which is comparing the water quality and microbial ecology of an urban and a rural stream. So why conduct this study? Well, prior literature suggests that urbanization is strongly associated with degraded stream water quality, and more specifically, the percentage of a stream's watershed that is composed of surfaces that are impervious to water correlates strongly with elevated water quality parameters such as E. coli and total coliform counts, alkalinity and hardness, and conductivity, just to name a few. However, these increased elevated levels of those water quality parameters, the effects of those on microbial community composition in these streams is far less understood. So in order to conduct this study, we selected two streams within the Greater Lake Erie watershed, Mill Creek and 12 Mile Creek. Mill Creek is a 19-mile long tributary to Lake Erie that has a 2-mile diversion under the city of Erie and has a very highly urbanized watershed. It starts up by I-90 and then comes down past the zoo before being diverted underground and eventually emerging by the wastewater treatment plant and emptying into Presque Bay on the bayfront by Ordock Road. 12 Mile Creek, on the other hand, is a far more rural watershed, just a couple miles outside of northeast PA, with a watershed that's mostly composed of agriculture and rural housing. And this map is a map of all the subwatersheds on the Pennsylvania Lake Erie border. And Mill Creek is here in the middle, denoted by a star, and 12 Mile Creek also by a star. And 12 Mile Creek was also selected for this study because it is comparable in watershed size as well as length to Mill Creek. So we hypothesized that we would find evidence of reduced water quality in Mill Creek when compared to 12 Mile Creek, and as a result we would see differences in the microbial community composition in each of the creeks, as well as we would likely find differences between sites along the same creek. So these are this is a map of all the sites that I tested. The Mill Creek sites were inherited from a previous study done on Mill Creek to assess the water quality before and after the underground diversion via the Mill Creek tube, as well as water quality at the mouth, the headwaters, and where Mill Creek intersects with a tributary. So I selected 5 sites from 12 Mile Creek to mirror these to the best of my ability, mainly focusing on the mouth of 12 Mile Creek, where 12 Mile meets with a tributary, and the headwaters of 12 Mile. So surface water samples were collected from all 5 sites weekly, as well as immediately after emptying into the bay and lake respectively. This was done via a mixture of grab sampling with a 500 milliliter bottle that you see in this picture, or via a Van Dorn water sampler, where the creeks weren't readily accessible by hand and foot. pH testing was also performed at every site in the field via the use of a Fisher brand Acumet portable laboratory pH meter with a 3-point calibration of pHs at 2, 7, and 14. Once samples were collected, they were stored on ice until able to be returned to the lab, where they were analyzed for hardness, alkalinity, and nutrients via the use of Hawk reagent tests, powder pillows, and spectrophotometry. The nutrient tests, phosphate and nitrate, were provided a colorimetric change that was measurable by the spectrophotometer to give us an estimate of the concentration that was within the water sample for nitrate and phosphate, respectively. Additionally, water samples were vacuum filtered for each site through a Wattman millipore microfilter paper that would catch the microbes on the filter paper and be ready for plating and for E. coli and total coliform counts, as well as being saved for DNA extraction later. And conductivity was also tested in the lab via the use of a Fisher brand conductivity meter. So now we're going to get into some of the results. These are the E. coli and total coliform counts for Mill Creek and 12 Mile Creek. These counts were estimated by, as I said, plating filter papers onto Petri dishes with pads that are treated with 2 milliliters of M. coli blue broth, which allows a color change in the colonies that grow on them. Blue colonies are E. coli, and the colonies that are red, it's kind of hard to see on the screen, are total or other coliforms. We filtered a 1 milliliter sample of water and a 10 milliliter sample of water through a filter paper for each site, multiplied the counts, the resulting counts, by 100 for the 1 milliliter sample and by 10 for the 10 milliliter sample, and then took the geometric mean of those two to give us an estimation of the E. coli and total coliform at each site. And then for each site, those were then averaged to give us a general picture of the E. coli concentration in the entire creek. So as you can see, E. coli and total coliform were both experienced elevated levels in Mill Creek when compared to 12 Mile Creek, and this could be because the amount of impervious surfaces, as I mentioned before, in Mill Creek prevents any sorts of fecal matter or any sewage runoff on the road from being filtered through the soil before entering the water body directly, or could be more suggest severe stormwater runoff as a result of the impervious surfaces experienced in an urbanized watershed. Getting into the nutrient results, interestingly enough, nitrate concentration was found to be significantly higher in 12 Mile Creek when compared to Mill Creek, and this is likely due to the use of fertilizers. As I said, 12 Mile Creek's watershed is far more rural and focused on farming, so perhaps the fertilizers used by the farmers out there are high in nitrate concentration, which then run off into the creek. But kind of perplexingly, phosphate concentrations experienced no significant difference between the two creeks, whether that means that maybe the fertilizers that the farmers in 12 Mile Creek's watershed use are not high in phosphate levels, or maybe lawn fertilizer levels of phosphate in the urban watershed are elevated, making it so that there's no significant difference between the two. Moving on to alkalinity and hardness, again, both found in elevated levels in Mill Creek when compared to 12 Mile Creek. This is likely due to runoff from construction projects using calcium carbonate as a component of concrete and other work, dust drawn up by drilling, etc., can all lead into or be runoff into the water. And since calcium carbonate is a major constituent in providing calcium for hardness levels and carbonate species for alkalinity. I think it's a fair assumption to make that there is a strong correlation between the urbanization and elevated hardness and alkalinity levels. And moving on to pH and conductivity, there is no significant difference between pH between the two creeks. They also fell in within the typical range of like 6.5 to 8.5 for a stream of pH. And then conductivity, on the other hand, was extremely elevated in Mill Creek, which is the total concentration of ions in the water. Granted, these 3 samples were taken during the winter, where road salt is spread extremely heavily throughout downtown Erie, as you may know. So I would like to get out in these next few weeks a few more times to be able to get a better picture of conductivity in Mill Creek when it's not the winter season. So that kind of wraps up the physical water quality characteristics side of things. Now I'd like to get into the microbial analysis. So as I said, samples were collected, filtered, and then stored in the freezer at negative 20 degrees Celsius until DNA extraction was possible. And for DNA extraction, we used the Zymo Labs Research Quick DNA Fecal or Soil DNA Extraction Kit, and we altered according to their protocol our process just for water samples, which essentially the only alteration we had to make was to break up the filter papers before starting the process. And the DNA, the Zymo Research DNA extraction method essentially boiled down to 4 major steps, which was cell lysis, isolation of the DNA, filtration of the DNA, and then elution to get DNA samples that are ready for PCR and downstream analysis. Speaking of PCR, we are going to send these we sent our DNA extraction samples just a week ago to Biomarker Technologies down in North Carolina, where they will be doing PCR and then 16S rRNA gene sequencing, which is a highly conserved region of the microbial genome, which will allow us to get the percent composition, relative abundance of bacteria in each sample. So then we will divide the genomic data into different, by class, family, etc., to compare the data between the 2 creeks, and we will try to assess whether the environmental factors, differences between the 2 creeks will cause a detectable shift in dominant taxa, decrease in ecologically important groups, or a potential increase in pollution-associated bacteria across creeks. So in conclusion, our findings align with our hypothesis that urban streams tend to have a lower overall water quality than rural streams, but bacterial community analysis still needs to be conducted to elucidate the relationship between water quality characteristics and microbial community composition. Thank you. Any questions?
Did you enjoy this study?
Yeah, it's been fun. I get to go outside. It's nice. Yep.
Did you test the salinity of the water when you were looking at the conductivity?
Not directly salinity.
So you're mentioning the difference in land use between the 2 watersheds, one being rural with farmers maybe using fertilizers and different concentrations of chemicals. Do you know in the rural dominated area, are those mostly farms that have cattle or other animals, or are they farms for plants and vegetables and stuff?
It's mostly grape farms,
Okay.
wine country out there.
Was any of your data influenced by weather, do you think? Like, were you sampling during times of drought or rain events?
need to assess that. I have the background data still, but yeah, I think there would be definitely an increase in probably all of those water quality parameters that I mentioned after rain events when compared to dry spells due to the increased runoff.
Questionnaire to know the type of fertilizer they are using and how frequent they are using it.
That would be a good idea. I have not kind of interviewed any farmers, but I think that would actually be a good step into kind of assessing why we see the discrepancy in nitrate levels and not phosphate levels.
Do you know what kind of salt the communities use on the roads? Could it be a carbonate salt instead of chloride?
I actually don't know, but I think, yeah, that's a good idea. I should look into that.
I'm just thinking, if you see more of nitrate than phosphate, is it because maybe phosphorus is attached to the sediment and it's not readily available in water, something like that?
That could be, but I think, or it also could be maybe the soils out in Northeast are nitrogen deficient, so they're treating the with nitrate, nitrogen-based fertilizer. But that's also another explanation that I could explore. Yeah.
Which stream had the high phosphate that was in this stream?
There was no significant difference in phosphate between the two creeks. 12 Mile Creek had higher nitrate, which fertilizer likely explanation, but Mill Creek and 12 Mile Creek not being statistically different in phosphate levels was kind of intriguing. Awesome, thank you guys.
All right, we are going to go ahead and get into our final talk for the session, and that is Tyler Brooks with the Regional Science Consortium. Hello.
Hello everyone, I'm Tyler Brooks with the Regional Science Consortium, and we'll be talking about changes in pollinator communities in newly restored dune habitat. So we had to restore a dune on Beach 8. There had been some issues with it blowing into the parking lot, and a weather event had completely destroyed an entire dune. So we had to bring in tons of sand and replace it, but if you don't plant lots of native species to hold that dune down, then it'll just keep blowing into the parking lot. So we had to restore the dune, and we thought it'd be interesting to see which pollinators would flock to a new habitat on the dune, while also using a dune with existing plants on it as kind of a control. If we look here, we can see these are the plants that we found on the middle dune. It represents 17 different orders. Dunes represent a unique habitat. There's a couple species up here that are state listed, such as hop tree and western poison ivy. It's one of the few places you can see either of those species in the state, as well as wild 4 o'clock. But the single dune had over 30 species and 17 different orders. It's something, oh, I said that already. So to monitor the pollinators, we had to put up blue vein traps, which we can see here. There's 7 of them on each dune. Insects are attracted to different colors. Butterflies and moths prefer like reds, and flies prefer white. So blue, we were trying to target bees specifically. And these are filled with soapy water, so the insect will go to land on what it thinks is a flower and get funneled down and get trapped in the soap and die. And then we would come and check them twice a week, remove any insects that we had, and wash the soap off of them and store them in alcohol for later identification. So in the blue vein trap, we can see it was mostly flies, even though we were trying to target bees. That might be due to flies are a lot more polyphagous. They'll eat a lot of different things, so they can take over these habitats that aren't necessarily covered in flowers, like Presque or the dunes. Especially earlier in the year, we saw a lot more flies than we did later in the year. And another major contributor to flies was midges. There's a huge midge outbreak this year. So that made up about a third of all flies were midges, which do pollinate indirectly. They don't eat the pollen, but they land on flowers. We can see that beetles and bees both made up roughly a quarter of the pie chart, followed by moths and butterflies, and then hemipterans, which are like stink bugs mostly, as well as psyllids, which are an aphid relative, spiders, and other orders such as bark lice. Here we can see a makeup of the flies we found in the trap, as well as a couple specimens. As I said, midges made up a significant portion of the population. Another group that's extremely important is these flower flies down here. Not a lot of people know them. A lot of people think they're a type of sweat bee, but these are flies that mimic bees, and they're extremely important pollinators, and a lot of native species of plant rely exclusively on flower flies to pollinate them. Then we can see three other superfamilies made up a large portion. There's the muscoids, which are houseflies. There's ostroids, which are bot flies, and then there's fruit flies, and they made up a significant portion of the group. Fruit flies tended to be, there's a lot more of them earlier in the year when there was nothing else pollinating, because they're polyphagists, so they eat whatever is really out in March and April. And then they kind of came down later in the year. The syrphids came up later in the year. And fly identification is hard, and there's a lot of groups that are hard to get down to just family without counting hairs on the sides of the body or genital examination. So there's a significant portion that we still have to tackle. You can see at the top there's a midge, and on the left you can see all the hairs on this botfly. And whether or not there's one or two bald spots for hairs can differentiate species. Beetles made up a significant portion. We can see a couple here. Scarabs, mainly June beetles, made up the largest portion. A lot of those are going to be hard to get to species because they also require dissection. Staphylinids, those are your rove beetles. Over half of the rove beetles that were collected in the trap were these flying burrowing beetles, which eat primarily dead things. So they might have been attracted to the dead insects that were already in the traps, or they might have been pollinating as a secondary food source. And then a lot of other groups you can see on the bottom here. This is Bassarius mammifer. It is a poison ivy expert or specialist. So it's a species that's primarily found on the dunes because it eats western poison ivy. Hence the bright colors to warn predators. On the top we have a June beetle. That's an invasive species of June beetle. And some other notable groups are ladybugs. Cottonwood is a major species on the park, a major species on the dune, and there's aphids all over them. And species that eat aphids are ladybugs. So all of our ladybugs were Asian lady beetle or white or pink spotted lady beetles. Beyond that, we have fireflies, weevils, jewel beetles, and a couple other smaller families making up the dune. And then our bees, this is our main focus. Usually when people see a bee, they just think of the honeybee, which is down here, which did make up a large portion of our samples, but wasn't the main species. The major species we saw were silky sweat bees. Other things we saw were halictids and megachilids, which are leafcutter bees, and mason bees, and potter bees, and square-headed bees. All sorts of species that people don't really consider and think about. Primarily these are what we're trying to track, so it was nice to see a large variety of them. This is another group that that is hard to get to species, but doesn't necessarily require dissection, particularly because it's mostly females. And then there's some other non-bee hymenopterans we got, such as a couple ants. I think there's 4 ants, as well as up here we have Microdonta, or Microbembax monodonta, which is a square-headed wasp, which is a sister group to all bees. And we also got ichneumonids, which are parasitoid wasps, which usually require DNA work to identify. Moving on from here, the project is still ongoing. As you can tell, we don't have a slide for LEPs yet, which is moths and butterflies. And as it's going, we still have the traps up. We're still checking them bi-weekly. Identification to species is something we'd like to get. A lot of this is still preliminary, getting things to order. And then we still have to compare our data from the restored dunes to the existing dune to see which species are coming to the new dunes. Like I said before, it's primarily flies coming to the new dune, but wasps and bees are secondary coming to it. And then we have until next July, we're going to continue monitoring them. And I'd like to thank all these people for helping. Sorry I ran through that. Does anyone have any questions? No questions? Yeah.
So once you collect the specimens and bring them back to the lab, what do you have to do to process them or leave them?
Once we bring them back, each specimen is stored in a container of alcohol, and they're stored by which trap they came in, which dune they came off of, what day it was collected. And then as we go forward, we're going to be able to tell which species need dissected and which species don't, and try to tackle the non-dissected species first. And then maybe if we have time, we'll get into the fruit flies and June beetles that need their genitalia examined for species identification. And then once they're processed, we can pin them. We'll have educational drawers. We'll have extras go to the museum. Things like that, whatever we can do to salvage dead specimens.
Was there anything you saw, like that you collected from the traps that just surprised you that you really didn't think?
Like I said, I didn't expect those like burrowing beetles, which primarily eat dead things, to be in the traps. And then I was shocked by just the amount of flies, even though they're only supposed to be targeting white flowers. They're in these blue vein traps for some reason.
Well, thank you to all of our presenters. That does conclude this session. We are going to take a break until about 2:40, at which part we will reconvene in this room for the final session of the day. So thank you, everyone.
Yeah. Yo.
afternoon, everyone, and welcome to the last session for today. We are happy that you stick around with us. My name is Mumi Femi. I'm a professor at the University of Pittsburgh, Bradford. So yeah, we'll call our first presenter today. Well, unfortunately, the first one we had to wait here. She didn't show up. She's not feeling fine. So the second person will go. And the second person is talking about assessing bacterial proliferation in swim bags over time. And that is Madison Foote from the University of Pittsburgh at Bradford.
Alrighty, so as she introduced, my project I'm going to be working on over the next 4 or so months is assessing bacteria proliferation over time in swim bags. So bacteria growth depends on environmental factors like temperature, moisture, and light. Swim bags often remain damp after practices and are typically poorly ventilated, creating favorable growth conditions for things like bacteria. This study aims to examine how bacteria colony sizes change throughout different points in the season. So my hypothesis is that as the swim season progresses, bacteria colony size is expected to also increase due to repeated exposure to moisture and limited drying conditions inside the swim bag. The goals of my project are to assess environmental influence, measure bacteria growth over time, test how bacteria grows throughout the season, and promote hygiene awareness. Samples will be collected from 6 swim bags over a 4-month period, with 2 samples taken from every bag during every sampling event. I'm looking to start swabbing within the next 2 weeks. A swim bag not in use during the season will serve as the control for the duration of my study, and the swim bag will also be sanitized before every swab is taken. Swabs will be taken from the interior bottom surface of each swim bag. The collected samples will be transferred onto growth plates and incubated under standardatory laboratory conditions. Following incubation, bacterial colony growth will be assessed by measuring the area of surface coverage. Colony size and density will be compared across the 4 sampling periods. Photographs will be taken, and a comparative graph will be generated to visually represent differences in bacteria growth among samples. Bacteria identification will be conducted using gram staining techniques as well. My expected results are bacteria colony sizes are expected to increase over time, with the largest colonies appearing in samples 3 and 4, because at that point we're pretty far into the season. January would be sample 3 and February will be sample 4, so the bags will have more time to basically just sit with wet towels in them, because it's typically what happens. This supports the hypothesis previously stated that persistent moisture and poor ventilation promotes bacteria growth. Some of my possible limitations would be variations in swimmer hygiene, like if someone per se is really good and cleans their bag after every meet, I'm going to have a different result compared to if someone just lets wet towels sit in there over the whole season. And frequency of use of the bag could also cause inconsistent results, because I know some people use the bag every day, some people only use it for meets. There's some limitation there. Like I said before, regular inconsistent cleaning of swim bags could significantly affect bacteria colony size and growth trends, and then differences in swabbing methods or surface area samples could produce varied results, though my plan is to swab the bottom interior of the bag, and possibly we have been talking about also swabbing a couple different areas of the bag. The implication of the project implies that proper hygiene and drying practices of swim gear can reduce bacteria infestation and promote the health of swimmers. And then my conclusion, the study aims to prove that bacteria growth is influenced by moisture and limited ventilation. Results are expected to show an increase of bacteria colony size as the season progresses. Regular cleaning and drying can reduce bacteria growth rate. Overall, the study hopes to show how proper hygiene and drying practices can reduce bacteria buildup in sports gear, improving athlete health and reducing odor or infections. Any questions? including, like for your sampling, are you going to be picking people from random from the team, or?
Honestly, it's probably going to be off a volunteer basis.
Yeah. Have you considered maybe including like a questionnaire about like how often they clean their bag and maybe like
No,
what
but
type
that's a
of
good
bag
idea.
they use?
Yeah.
Yeah.
Are you gonna use like a control of just like normal water, considering there's like chlorine in pool water?
Can you repeat that question?
Like, there's like chlorine in pool
Yeah.
water. So, are you, like, would there be like a control, like, let's say with just like fresh water?
I haven't really thought about that. So at this moment, no. It's just straight chlorine and what the bags, yeah.
Kind of going off what you said about the chlorine, are there specific bacteria that you're going to be looking for?
That I haven't quite figured out yet. Right now we're just, like, we know, like anybody knows if you leave a wet towel in the bag, mold's gonna grow. So it's kind of just, we're kind of just seeing what we pick up off the swabs to view under gram staining and look under the microscope to kind of see what it is, and then we are gonna identify the bacteria.
So do people does every member of the team have the same bag? Like, are they matching
Yeah.
set? Okay, I was curious
Yeah.
if there's variation in the bags like that.
No,
kind of
they
thing.
are
Okay.
all the same,
Okay.
same bag, same everything.
How do you guys wash the bag? That's kind of a big thing as well.
Well, you can put them straight in the washer, or you can disinfect them with like spray and wipe them down. There's a couple different ways you can clean them.
I know for a personal experience, the school washers, I've had like 3 bags in my washers. They're not
Yeah.
great bags.
Yeah.
Do you think chlorine will play a role in preventing any bacteria growth, or?
See, I don't think so. Just because I know from experience that they'll grow mold. They can get really gross really fast. Okay.
Okay, we have the first one here. Okay. All right, so we will go to the second presenter for today. All right, our next presenter will be talking to us about assessing lead paint exposure in campus building using XRF. Let's have Elizabeth and Madeline.
Hi everyone, my name is Madeline Crew.
And I'm Elizabeth Kibler.
And we're both students of environmental science at Gannon University. We've been working on this project all semester and has been assessing lead paint exposure in campus buildings using XRF analysis. To get into what we're going to talk to you about today, we're going to be giving you some background on what is lead and historical data and health impacts that it brings, and then going into our research goals and methods, and then we'll share our results, our interpretations of those results, and the future experiments we've planned. And then we'll give some recommendations off of the risks and impacts.
So to start, why this is important. So lead is a naturally occurring heavy metal, and it is toxic to humans in higher concentrations. And so it's important to identify it and its presence in campus and the exposure routes. So our main focus is adults, as we're all students and faculty are adults, but frequently children are the most at risk for experiencing the health effects of lead. So lead is most commonly exposed in today's day and age as dust from paint chips, because paint was in lead for until 1978. It can also be found in the soil because it was also in gas, and any car exhaust that mixed with soil, lead has remained persistent in the environment. And it can also be found in drinking water due to the corrosion of metal pipes that contain lead. It can also be found in some old chinas and in lead bullets if anyone is consuming game that they hunted with. So for health effects, focusing on adults, it can have cardiovascular effects, including increased hypertension, neurological effects like memory loss, fatigue, brain fog, and some mood changes, and difficulty in decision making. In children, it can cause death, but through like renal failure and neurological issues as it affects the nervous system. So this is a photo that we took from during our research in an on-campus building. So you can see that this paint is clearly chipping and mixing in with the carpet, and this was lead-positive paint. So, like I said, the most common exposure route is through chipping and dust. So lead paint is still prevalent despite it being banned in 1978 for residential use in paint, and despite the fact that it was banned in gasoline, it still persists in our environment today. According to the EPA, 87% of homes built before 1940 contain lead paint, and 24% built between 1960 and before its banning in 1978 contain lead-based paint. And 72% of homes in Erie were built before 1978, which means that there's a high risk that these homes are contaminated with lead. And Erie was ranked number 27 in a study of most lead-contaminated homes in the US.
All right, so now to get into our research goals and methods. Like Libby said, when we began our project, we had to take into account that Erie the buildings in it are very dated. So thinking about this, we decided the best thing for us to do was to test as many campus buildings as possible to gain as much of a grasp as the risks that could be present there. We were able to move into distinguishing the risks present by analyzing the duration of exposure. For example, a workplace is going to have a different amount of exposure than a living space because of the dust chipping and just being exposed to it. And moving further into our research, our main goal is to bring awareness to the toxicant altogether. Libby and I were lucky enough to have courses that really showed us the toxicant and the ugly side of it, but we know that's not the same for everybody, so we just want to bring that knowledge to everyone. Through our research, we've been using one main method of detection, and it's a Hitachi Science handheld XRF analyzer. XRF stands for X-ray fluorescence, and it's a non-destructive analytical technique that allows us to detect lead without causing any damage to the test site itself. It works by exciting atoms with X-rays, causing them to emit secondary X-rays from their inner shells, whose energy reveals the elemental composition of them. We've been able to make it to 8 on-campus buildings, 3 of them being office spaces and 5 being residency locations to gather data.
So for our results, we found, well, first to identify what a positive result constitutes, it would be a reading of lead greater than 1 milligram per centimeter squared. And this was data that the gun itself would give to us. We found that 7 of the 8 buildings had positive results. All 3 office buildings had positive results, and 4 of the 5 residential areas had positive results. So these were our results. We grouped them by what type of material that the paint was on. So our most prevalent was on windowsills, sashes, and frames, followed by the trims, like wooden baseboards, door trims, and frames. And I'll show you, this graph is a little better to see the distribution here. And here's some photos of some of these positive results. So these are 3 of the very frequent lead paint places, so windowsills and then a door trim at the end. This is us actively using the XRF technology. So from these results, we looked at them and we tried to notice any patterns that we saw. Because it was in all buildings, we had to look at what types of materials were showing up as positive. So we found that lead was commonly found on wooden surfaces, which included the trims, the baseboards, and windowsills, window sashes. Those were typically wooden materials that the paint was on, rather than finding any on plaster or drywall. It was more common that we were finding it on wood. And we also found that in a building that had multiple floors. Sometimes the first floor would have been renovated more recently and we weren't finding any positive results. But on the second and third unrenovated floors, there was frequently positive results. So from that, we were analyzing why wood may be a surface that frequently has positive results. So we were thinking and hypothesizing that there might be weathering of the paint through moisture, temperature, and microorganisms, as well as weathering on these windowsills through UV light. And we're hypothesizing and discussing whether this was weathering of a non-lead-based paint coating over a lead-based paint. Because it was banned in 1978, it's likely that these places have been repainted since then. However, most of the time the lead wasn't scraped up first. They just slapped a few more coats of paint on top of it. So we think that there could have been degradation of this non-lead-based paint to expose the lead-based paint beneath it. And this would still cause health effects. And so we think that the wood was more susceptible to this weathering and degradation than plaster or concrete drywall.
So now moving into some of our recommendations. We looked at exposure in housing and workplace exposure and their differences. The duration of the school year spent living and sleeping in rooms with lead-based paint on campus, and it exposes, in the places we tested, we said it exposes about 40 students per year. And workplace exposure includes 40 hours a week spent in the workplace. So over 2,000 hours spent per year exposed to lead paint. And exposure does not only occur through contact, like some people may think. The main path of lead exposure in the living and workplace is inhalation, because when lead paint does become that dust, it can be easily taken up into, but the fine particles can become part of the air we breathe day to day. So there is risk with spending each day living and working around any lead concentration. And for further action, we just recommend stripping the paint and disposing of it properly, testing the wood, repainting, and we are going to go along with future experimentation to test the effects of UV light on paint and possible wood degradation. Thank you.
Questions for that?
So you mentioned inhalation being the most common exposure pathway. Like, at what concentration does lead become dangerous to inhale?
I don't know the exact concentration, I just know that lead bioaccumulates in our bodies over time, so it matters what level it reaches in you.
Infrequent and chronic exposure through working at the same desk for 30 years, every single day, day in and day out.
Build up.
Yeah.
Build up.
Did you focus, like for your locations that you chose, did you intentionally try to pick the older buildings or like the older residents?
So yes, but luckily a lot of the workplaces are old, like just in general.
Yeah.
The living places, the one that we tested that didn't have any positive readings is old as well, but it must have been renovated in recenter times, but.
We chose locations that housed less students because they were less frequently redone. So we actually looked at the sorority houses on campus, which are part of campus property. Because we had both lived in these freshman dorms that housed like hundreds of kids, and those are frequently redone. They require more maintenance, and they just get worn through more frequently. So we looked at houses that have less students, but obviously their lives matter too, and their health matters. But they're less likely to have been redone recently, and we found that to be the case.
So you told us about the limit of detection of the instrument, but do you do any quantitative, the quantity of the lead remaining inside the paint? Do you have an idea of it?
So our instrument reaches a threshold, and we haven't taken anything back to the lab to get real data and like numbers on it, because then that would require us damaging the site and people are living there, and that would be a little too invasive right now. So what matters to us right now is that there is lead present.
Yeah, and the positive threshold was that 1 milligram per centimeter squared. We did have some locations that tested over 0 but under the threshold, which we took note of that, but they weren't included in like the positive results. And then the threshold, the highest number that we would hit was 5 milligrams per centimeter squared.
So pretty much a two-part question. One, does the instrument makes reading just on the surface, or the X-rays penetrate at depth in the layer of paint? And part two, do you think the thickness of the newer paint that covers the lead-based paint is going to have an influence on those readings?
Yeah, I think, like we were saying when we tested on something thicker, like we were in a basement and tested on like a concrete surface that had a very old looking paint on it, and it came back as negative. So, we kind of hypothesized that the thickness of this material, like you said, could have influenced its reading if like the rock or the concrete was able to maybe absorb more and not reflect as much. And when we do go back to the lab and test our future experiments with the wood, we'll take into account like different amounts of paint and what the threshold level of like coverage could be to prevent lead exposure, and thickness of the wood itself as well. So we don't have those answers yet for the most part, but we are we're questioning that as well.
Okay. If you check the permissible exposure limit for lead for individuals in the office, and you discover that what you're finding on the paints in your school is not up to that, should it still be a concern? For example, the PEL for lead, I think for 8 hours, but it should be around 50 microgram per cubic. So if you think yours is lesser than that, would this still be a concern that lead is, I know it's terrible, but then would that be a concern?
I think we do want to share these results with the university, and I think they could kind of use that logic to be why they shouldn't invest money in redoing these buildings. But I think for the health and safety of the individuals and their faculty, it would be of best interest to spend the money to replace this, although legally they may not have to. Personally, I think most people would feel comfortable if they knew that they weren't exposed to any amount of lead. However, if it's under limits, it could be allowed.
One more question for you guys that I thought of. So how many of the sites, because I know you had the photo of one that was clearly chipping paint on the baseboard. Were most of the sites like that where you clearly saw the chips around, or were like they kind of more intact?
Yes, in the housing places, a lot of them were like that actually. And actually one house we went to had a kitten like wandering around us while we were testing, and it was like nuzzling around on the floor, like could easily eat the chips. But yeah, that's very common, yeah.
Because a lot of these houses were older, like it just kind of old, thick paint that was kind of just coming up.
Yeah.
So very easily chipping off. And
yeah.
Give it up for them, guys.
Okay. All right. Oops.
Okay, we will have our last presenter for today, and that will be Isaiah, and he will be presenting molding campus building, environmental drivers, and mitigation approaches. You're welcome.
Good afternoon, everyone. My name is Isaiah Sanchez, and my capstone project is titled Mold Matters. To start, mold is a common part of our environment. It's found indoors and outdoors, and grows especially in damp, poor ventilated spaces. Older university buildings, like many here at Pitt Bradford, even though we're in Lake Erie, They have aging infrastructure and climate control systems that make them more vulnerable to moisture buildup. Health-wise, exposure to indoor mold can cause symptoms like respiratory irritation, headaches, fatigue, and allergies. For students, that can be a big impact on their daily comfort and learning performance. The CDC and NIOSH note that mold itself isn't rare, but when it thrives indoors, it can signal a moisture problem that must be addressed quickly. And that's exactly what my research focuses on. Identifying those conditions and testing ways to fix them. This image here is like a schematic of townhouses back in Pitt Bradford. This project began with what many students have seen or smelled firsthand: mold in the dorm bathrooms, the laundry rooms, and even shared spaces like the library. And some students have reported fatigue, headaches, and previous symptoms I mentioned in previous slides before. And they have been living in these spaces for long periods of time. What's interesting is that our housing contract actually acknowledges this risk. It includes CDC-based information explaining that mold can occur naturally and asks students to report any visible growth to the housing office. And that's good policy. But even with this awareness, mold just keeps reappearing. The current system focuses more on the response, not longevity on prevention. This is what motivated me to study mold conditions scientifically, to collect measurable data on humidity, ventilation, and spore presence, and to test low-cost ways to reduce the problem before it starts. My research objectives has 4 main goals: to measure mold levels and environmental factors like humidity and ventilation, to test simple, affordable interventions like portable dehumidifiers and clean treatments. These could be like bleach, vinegar, etc. And the third would be comparing conditions before and after those interventions. And lastly would be using the results to raise awareness and inform better maintenance policies for student housing. For the materials I'll be using for this ongoing experiment would be a spore cassette and an air pump to collect airborne spores and calculate spore concentration in the air. I'll also be using surface swab kits to detect mold on visible areas like vents or tiles. A hydrometer and thermometer will also be used to monitor humidity and temperature. And the DMAT, also known as a dampness and mold assessment tool, will be used to document signs of dampness and mold in buildings. During the intervention phase, I'll be using a portable dehumidifier and household cleaning agents, seen right there, like bleach, to test simple solutions. I'll also be wearing gloves, goggles, and N95 masks, just for safety and precaution to prevent contamination of samples. The 2 main hypotheses I'll be testing is buildings or rooms with humidity above 60% will show higher mold spore concentrations than well-ventilated rooms. And after introducing a dehumidifier or cleaning treatment, airborne spore counts will drop by at least 20% within 1 week. And here's a mock table for an example of what type of results will come. So this is a methodology of or flowchart of the first phase, where I'll be conducting field sampling in 2 to 3 campus buildings that have reported mold issues. which would be common buildings like the library or the student dormitories, such as like the townhouses. In each building, I'll be sampling 2 rooms per week for at least 2 weeks, potentially pushing on to maybe a month for longer testing. And I'll be recording the air spore counts, surface swabbing, humidity, temperature, and ventilation. And I'll also be conducting an anonymous survey asking students and faculty if they've noticed any mold, musty smells, or any symptoms like fatigue or respiratory irritation. This will help me link environmental data with people's lived experiences. In the second phase, I'll test low-cost interventions. In the same rooms, I'll be placing portable dehumidifiers in clean, visible mold areas using household cleaning agents. Then I'll repeat air and surface sampling for 48 to 72 hours later and again 1 week later. The goal is to see how much mold levels drop and whether conditions stay improved. This experiment focuses on practical solutions that could be realistically applied in student housing. Not expensive equipment, but everyday tools. I expect to see that rooms with higher humidity and poor ventilations have higher spore concentrations, supporting my first hypothesis. After using dehumidifiers or clean treatments, I hope to see a significant reduction in spores, at least 20% or more, supporting my second hypothesis. And with the survey's responses, I expect them to have a higher relationship with buildings with higher measured humidity. So I have an alternate laboratory experiment if access to dorms or building is limited. I'll be using a controlled laboratory simulation where growing mold cultures on agar plates under different humidity, temperature, and light conditions. This can help verify how those environment variables affect mold growth rates in a controlled setting. So this project builds on the university's existing house policy, which already acknowledges mold risk and references the CDC guideline. My research intends to add data, interns, reports, and maintenance complaints into measurable and environmental evidence. These results can help housing and facilities move from reacting to complaints to preventing a problem as a whole. By identifying high-risk areas before mold reappears, the university could improve air quality, update maintenance schedules, and even revise the housing contract to focus less on disclaimers and more on prevention. Beyond the campus, this contributes to broader indoor air quality research and higher education, a field that's still, it's being informed to a lot of people, but the actions are not done. It's worth noting that the CDC and the NISH do not recommend routine air sampling for mold because there is no universal health-based limits. However, they do emphasize moisture control and environmental monitoring. My experiment aligns with that guidance. It doesn't try to diagnose illness, but instead focuses on environmental prevention. So, in conclusion, mold exposure is student housing. It's not just a maintenance issue, but it's an environmental health issue. And this project just aims to measure, to test, and prevent mold growth through practical evidence-based strategies that campuses can afford. The long-term goal is a safer, healthier living environment for students and staff where air quality is maintained proactively, not reactively. Because at the end of the day, healthier spaces support healthier students. And these are key references supporting my research.
dorms on campus, like, because I live in a dorm on Bradford, so like, do you plan on going around like different dorms and surveying and seeing what's happening, or?
Yes. I intend on going to like certain dorms and asking permissions first to see whether or not I'm able to conduct these tests. I do have to contact housing and talk to anyone that does live in a dorm to make sure the privacy is respected.
Yeah, you should definitely go to a dorm.
Maybe just some other sites to consider are ones that aren't obvious. It seems like mold builds up in air vents, and then if you turn on the air conditioner or heater, it blows the spores out. Or sometimes on the filters of, you know, different vents where like HVAC systems, you know, that can distribute it over a wide range. That might just be something to look into that isn't really obvious for somebody that's living there, like seeing it on a ceiling tile.
I know you said you were going to concentrate on 2 rooms in each sample site. Are you going to focus on rooms that have a lot of like water exposure, like bathrooms? Or are you going to try to do like a little of each? Like you wouldn't expect as much, say, in a bedroom, but you would more in a bathroom? Like what are you thinking as far as that?
I intend on doing like the bathrooms because the showers there is where most of the water will be. And some bathrooms in the dorms are known to have like little to no ventilation, which has caused instances of mold growth. And I intend on doing like the living room, as it's where the space is connected to like the kitchen in our dormitories. And that's where majority of people put their foods, or they're using like sinks and everything.
Okay, thank you so much. All right, we have come to the end of the presentations for today. Thank you for coming around, and we hope to see you tomorrow morning, guys. All right, thank you. Give yourself a round of applause for being here.